Okay, there we go. Okay, I was worried about it for a second. Hello and welcome, my gentle and of course very modern apes. I hope that everybody can hear me. I'm going to just assume that you can, unless somebody tells me otherwise, because I've kind of got an eye on the chat here. Welcome to the, fifth of these episodes of this series where Will and I are sort of working through the support for evolutionary theory. Will being a pastor from Colorado, who's also notorious in a good way or a bad way, depending on who you're talking to, for the final experiment, an experiment that took several globers as well as flat earthers to Antarctica to see the 24-hour sun. The purpose of this series, for those of you who may not know, if you've maybe sort of wandered your way into this stream, is Will is a creationist, and he's a young earth creationist specifically. And so he's sort of taken this plunge to learn what evolutionary theory is and what the support for it is as well. Like, why do people seem to be convinced by this idea? Sort of tagging along with that is this idea of the ancient age of the earth. So Will and I, once a month for 5 months now, this will be the 5th month, have been sitting down and he's just letting me lecture at him. It's really a dream come true for me. Because this is the 5th, we've actually finished our first sort of unit, right? Because there's a lecture for every month, and we sort of have 4 in the initial unit, which is basics of evolutionary theory and the age of the earth. And then the second unit is sort of case studies in evolutionary theory. And so we're sort of starting our first series of case studies as we move from the present day back through deep time, going through the support for evolution from genetics and paleontology and ontogeny and comparative morphology and things of that nature. So that's what we're going to be doing today. And I'm super duper excited to do so. Will, how are you?
I am great. I am great. Thank you. You've put me on quite the research mode, which has been good. It really is. It's exhausting, but I love it.
Yeah, I get to be in my bonnet about topics every once in a while as well, and it sends me down like a rabbit trail. Like, actually, today we're talking about cetacean evolution, so this is a perfect example. I reread a couple of different textbooks that I have, as well as this little book here that's actually just conveniently still sitting here by Hans Thewissen, called The Walking Whales, on whale evolution. And because the support for whale evolution comes from so many different places, I've had to like really learn a ton about this super weird group of animals. And so it like, for a couple of the sources and even a couple of the pictures that are in this slideshow, it was like 3 hours in the making of me trying to find, okay, I need this specific reference photo of this toe of this specific proto-seeded whale so that I can show a side-by-side and you can see sort of the point that I'm trying to make. And I've learned a whole lot as well. So this has been, and hopefully will continue to be, a wonderful learning experience for me too. So awesome. Normally, actually 2 announcements. Well, 1 major announcement, and then we'll get into this sort of lead into the show for today. Because for those of you who don't know, the way that we begin these shows is Will give a sort of recap from the last lecture, where he'll talk a little bit about the things he agreed with, the things that he disagreed with, and tell us a little bit about where he's at. I'm super excited to hear about where he's at today, because we covered Age of the Earth stuff last time. And that's kind of I would argue, I said this at the beginning of the last lecture, but like a linchpin for this concept, at least of young earth creationism versus evolution, because evolution requires a lot of time. And conversely, young earth creationism would insist that that time doesn't exist. So we've got sort of these 2 opposing hypotheses, and I'm eager to hear what Will thought of that lecture series, because I've intentionally, I told him this before we started the show, I've intentionally not asked. I'm going to be just as surprised as you guys are as to where he's at. Obviously, I've got my fingers crossed because I'm biased, but wherever he's at, that's the learning process, baby. So wherever he is going to be good by me, and hopefully if he has any questions, I'll actually be able to answer them. The other thing of note is that super chats for these shows. So if you want to super chat in and ask me a question or ask Will a question, you're free to do so. All revenue from super chats goes to 2 specific charities or nonprofits that Will and I have picked. The super chat money gets divided in half and sent into these 2 charities. It has not yet been done because, as I was complaining to Will beforehand, it's really, really hard to actually calculate how much super chat money each stream makes individually for reasons that are completely unbeknownst to me. So I have to sit down and do it by hand, which sucks. But our 2 charities are, Will has picked Exodus Road, yes, if memory serves, which is helping people out of human trafficking style situations. And the one that I've picked is Pro Literacy World. So I'm doing a literacy initiative to help adults regardless of where you are, learn how to read and learn how to critically think as they sort of read. I think that's a prerequisite, obviously, for education, the type of education that we're doing today. So it seemed like a really good choice. And primarily I picked it because it's got a phenomenal track record. So those receipts will be up the second I get all of this stuff tallied. I think that's it. I think that's all I have. Will, can I bop over to you to give your summary from last time? And I'll be taking my notes as usual over here.
Yes, definitely.
Solid.
Go ahead and pull up my screen here.
You are added to the stage, my friend.
Okay, let's do it. All right, so this picture is super fun for me. I'm actually in the Hell Creek Formation here, digging for dinosaur bones. And this was the first dinosaur bone that I found. I was with my daughter, and so I had her take this picture. If you've never done this, it's an incredible experience. It's very hard to explain. These things are very fragile. You have no idea what you're going to find or not find. And so when you find something that's a confirmed bone, you get excited. You also don't know what it is. You don't know how far back it's going to go. All that gray stuff that you see at the top, all of that has to get removed. That brown layer below is where all of the dinosaur bones are. And so in the Q&A, I will tell you what I found, which is super exciting. But
good, because I was going to ask.
I thought this picture was appropriate for our conversation. Okay, so Age of the Earth. Now, before we get to my thoughts on Erica's last presentation on the Age of the Earth, I do have some pictures to share. Now, I don't have a lot. For those of you who missed the last session, I was actually looking for whales in the Western Atlantic around Puerto Rico. And I had, I would say, my least successful trip to date. One day, a friend of mine actually invited me to a part of the island on his boat. And I thought to myself, there's no whales at that part of the island. So I said no. And that whale was right next to his boat. And so I did not take this picture. He did. So good for him. The trip was so unsuccessful that I'm actually going to be returning to try again later this month. So hopefully after our cetacean session today, I'll have better pictures for everyone next time. I will share 2 other pictures I took for fun. This is one of my favorite spots on the island to look for whales. And I got this really cool heron picture. By the way, I'm not a photographer, nor am I an artist. Everything I take is with my cell phone, and I don't edit pictures. So everything you see is just as it is with my phone. And then another spot I love to look for whales. I took this picture. It is completely unedited. It is not cropped. I want to enter it into some sort of a competition or something. Once I took it, I couldn't believe how it came out. But this little gecko was on the wall. That's the sky in the background, and it just turned out perfect. So a couple of fun pictures for everyone to see. Okay, let's talk about the Age of the Earth. Full disclosure, I came into our last discussion on the Age of the Earth feeling confident that I would be able to engage with Erica's arguments. And this is because, unlike evolution, I've looked into the Age of the Earth previously. I was wrong. Okay, I was completely wrong. I was headed to bed after our last talk, and my wife noticed a defeated look on my face and asked me how it went. And I looked at her and said, Erica crushed it. That is a verbatim quote. So my initial conclusion is Erica's presentation on the Age of the Earth was the single best presentation on the topic that I've ever seen, hands down. So great job, Erica. Now, so the question is going to be this. So Will, have you changed your mind on the Age of the Earth? Yes, but before I elaborate on that, 2 things happened in the last few weeks that really impacted me and influenced my new position on the Age of the Earth. The first is I was made aware of a geologist who changed his own position from old Earth to young Earth. And number 2, Hugh Ross kept popping up in my life and led me down a very interesting rabbit trail. So let's start with Monty Fleming. He is a geologist. He has a PhD. He switched from old Earth to young Earth, and he details his descent from deep time in his book, Stories About Earth's History. There's a picture of it on Amazon if anybody wants to look into it. So this really shocked me that a PhD geologist would switch from old Earth to young Earth. And after Erica's incredible presentation, I just had to know why. So I decided to read his book because I was super curious what would cause such a change for someone who is actually an expert in geology. Now, his book covers a lot, but coastal erosion and the discovery of living organisms. These were the two topics that were the most interesting and shocking to me. So there's a lot more in the book, but these two were the biggest two. Let's start with coastal erosion. Here's a quote from page 10. If we take 8 inches per year as the average rate of erosion for the Pacific coast and 31 inches per year for the Atlantic coast and look forward in time, the two coasts will meet about 4.2 million years in the future near Salt Lake City, Utah. Also from page 10, the North American plate, the tectonic plate the United States calls home, supposedly formed 3 billion years ago, yet simple math and a few facts of life show that the ocean would have eroded it down to sea level in only 0.14% of that time. Now, this wasn't even the most shocking thing for me. What he said next is almost hard for me to believe. From page 10, the textbooks I studied for my MS and PhD in geology never mentioned this issue, and I've never seen a scientific article on the topic. That's stunning to me. Don't exactly know why, but that's big for me. Okay, up next is living organisms. From page 74. When researchers started looking in earnest for original soft tissue and other original material in preserved bones, they found it everywhere. All of these examples of ancient biomolecules shocked the scientific community, but the story gets even wilder. Scientists haven't just discovered tissue from dead creatures, they've discovered a number of living organisms from equally old deposits. I had never heard of this before. So they have found living bacteria in a 34,000-year-old core of salt from Death Valley, California. All the papers are linked there at the bottom. They found living bacteria in 500,000-year-old ice from Siberian permafrost in northeast Russia, in 112 to 121 million-year-old salt crystal from the Permian Salado Formation in southeast New Mexico, in 225 to 280 million-year-old salt from an alpine Permian salt deposit in Bad Ischl, Austria, and from 250 million-year-old salt crystals from the Permian Salado Formation in southeast New Mexico. Page 76, quote, 34,000 years is a problem. Based on everything we know about biology and chemistry, bacteria can't survive in a tiny pocket of supersaturated salt water for 34,000 years. These discoveries have sparked quite a bit of contention. Generally, the arguments have been most intense between those who make the discoveries and those who demand that such discoveries simply can't happen. This is new to me. I don't know much about it, but it's fascinating. Next is Hugh Ross. Hugh Ross is an astrophysicist. He has a PhD. He is an old Earth creationist, and he is the founder of an organization called Reasons to Believe. Now, Hugh Ross, for whatever reason, has been frequently popping up in my life recently, so much so that I could no longer keep ignoring him. First, he was in a documentary that was recently released called Universe Designed. This was made by an atheist who converted to theism based on the design he saw of the universe. Hugh Ross is in that documentary. Then he was in the documentary that was just released called The Case for Miracles from Lee Strobel. And then just in the last 2 weeks, this video popped up in my YouTube feed, which is Erica with Fuzz. Fuzz is with Reasons to Believe. This is a conversation Erica had with Hugh Ross's organization. It was really good. I think it was almost 2 hours long, so I watched that entire thing as well. So before I had a chance to look into Hugh Ross and Reasons to Believe, I wanted to understand the different categories of views on evolution and origins. I didn't exactly know what an old Earth creationist was, nor what a theistic evolutionist was. So this led me down a rabbit trail and another design project, LOL. What I did is I made a list of the 5 main views on origins and evolution. I did leave out intelligent design as they have pretty varied views within that camp, so it was way too confusing. I specifically, this is important, listed people who are credentialed and or brilliant and or I have a personal connection with. So that's important here. These people that I list for a reason. So the first category is atheistic evolutionist. By the way, when I say atheistic, I also mean agnostic. Then there's the theistic evolutionist. There's the old earth creationist, the young earth creationist, and finally the atheistic anti-evolution group, which most people may not know about. All right, first up, atheistic evolutionists. Obviously, we've got Erica. Then we have Aaron Ra, Aaron Nelson. I have a long history with Aaron Ra. It goes back many years. We've spoken many times. We've emailed. I like him. And then there's Grayson Hawk. So he's on YouTube. His channel's called Grayson Hawk. Grayson and I were actually going to tour the Creation Museum last summer together and kind of do a live on YouTube talking about the claims being made there. Due to my scheduling conflicts, we didn't have a chance to make it happen, but he's a really cool guy. I like Grayson. Then we have theistic evolutionists. First is William Lane Craig, who has an organization called Reasonable Faith. William Lane Craig is absolutely brilliant. He destroyed Sam Harris in a debate. That's one I'll never forget. Then we have John Walton of Wheaton College. Multiple people have told me about his book about Genesis 1, which I want to read. And then we have Clint Laidlaw. He's got a channel on YouTube called Clint's Reptiles. I love this guy. He's amazing, friendly, nice, great content. I watch as much of it as I can. Then we have old earth creationists. First is Hugh Ross, who we've been talking about with Reasons to Believe. Then we have John Lennox of Oxford University. I think he's the #1 Christian apologist of our time. And then Greg Kokel is a guy who has an organization called Stand to Reason. I've actually been influenced and impacted by his ministry since I was a teenager. So he's been going about this a long time. Then we have young earth creationists. First is Danny Faulkner. He's with Answers in Genesis. He's a PhD astronomer. Danny and I go way back. We first met well over 20 years ago. I was very young, didn't have kids yet. He tried to talk me into becoming an astronomer, and he almost succeeded. Then we've got Donnie Badinsky, who has a YouTube channel, Standing for Truth. Anybody that follows Erica's channel definitely knows who Donnie Badinsky is. They go back and forth quite a bit. And then we have Rebecca Davis. She runs a YouTube channel called Examining Origins. Her channel popped up in my YouTube feed some time ago, and I'm really glad that it did. She does an incredible job of being passionate about what she believes. She invites people who disagree to come on her channel. She admits when she's wrong. She has very cool, unique guests on. So the YouTube algorithm definitely helped me out there. Then we have Atheistic Anti-Evolution Group. First is David Berlinski. I mentioned that his quotes have stuck with me for 18 years from the movie Expelled. Then we have Dennis Noble, who I think a lot of you are probably familiar with, who used to teach at Oxford University. He rejects neo-Darwinism. And then finally, we have Bradley Monton, who used to teach at the University of Colorado. He is best known for his book, I think it's called Looking for God in Science. An atheist defends intelligent design. So anyway, I'm proud of what I created here. This took a long time. Not as proud as what I created last time, but I had an aha moment when I was looking at this, because every single person on here I respect. Every single person on here I have a personal connection with. But I had an aha moment, which is this. Almost all of them are wrong. So if Erica and Aaron Ra and Grayson Hawk are right, everyone else is wrong. If the theistic evolutionists are right, everyone else is wrong. If the old earth creationists are right, everyone else on here is wrong. If the young earthers are right, everyone else is wrong. And if the atheistic anti-evolution group is right, everyone else is wrong. So this was an aha moment for me. I'm looking at all of these brilliant men and women on one screen, all smarter than I will ever be, and most of them are flat out wrong. They're going to be wrong about evolution, or the age of the earth, or the existence of God, or some combination of those three. But why? That's always the question for me. Why are there so many different views? The only logical conclusion I could come to was that there must be valid arguments for each of these views. These brilliant men and women are not going to hold a view without evidence. That makes no sense. Herein lies the problem that I am personally guilty of. When you only consume information from just one view, you develop a belief that the evidence overwhelmingly supports your view and that the other views don't have much evidence. But that's just not true. There are valid and compelling arguments for every major view on evolution, the age of the earth, and the existence of God. Here are just a few of them. From astronomy, old Earth, distant starlight. This is a really good argument that the Earth and the universe are old. But from astronomy, there's a good argument for a young Earth, which is the faint young sun paradox. If you haven't heard of this, Google faint young sun. First thing that will come up is Wikipedia, the faint young sun paradox. On the right, you'll see it says it's an unsolved problem in astronomy. The second thing that will come up is a paper that's on NASA's website, which you can read. The highlight there at the bottom of the first paragraph says, as of now, there is no theory or even a credible scenario to resolve this issue. Just to sum it up for people if they haven't heard of it, our sun gets brighter the older it is. And so if we go back in time, deep time, it becomes very dim, and therefore the Earth would be cold and could not support life. From geology, old Earth, we have radiometric dating. Very convincing argument that the Earth is old. For a young Earth, we have coastal erosion, which I mentioned from Monty Fleming's book. From paleontology, for an old Earth, we have faunal succession. I've been thinking a lot about this, and I do think this is a good argument for old Earth and evolution, is this order of fossils that we find. But from paleontology, for a young Earth, we have original biological material from dinosaurs. Now, I do not have answers or rebuttals for the arguments Erica presented for an old Earth. I also do not have answers or rebuttals for the arguments presented for a young Earth, including the faint young sun problem, coastal erosion, and original biological material found in dinosaur bones. So I will admit, I'm more than willing to admit and concede that the quantity of arguments and data favor an old Earth. So please don't think that I think that this is somehow there's an equal number of arguments on each side. I don't. But at the same time, there are valid arguments for a young Earth that don't seem to have solid answers. So Will, have you changed your mind on the age of the Earth? Yes, I have. Going forward, if someone asks me how old the Earth is, I'm going to answer honestly, I don't know. That is the honest answer. And the reason I'm going to say I don't know is quite simple. There are people I trust and respect on both sides of this issue. They are all much smarter than me. And so I'm going to continue to consume content from both sides and continue to contemplate this question. Finally, I am very tempted to put together something similar to the final experiment concerning the age of the Earth. But these types of endeavors, and I'm speaking from personal experience, usually end up taking way more time than anticipated and also end up over budget by significant amounts. So we will see.
Awesome. I love that. And I love your pictures, and I took some notes, and there's some things that I'm going to need to look into. For instance, okay, let me just start at the top. First of all, I'm really sorry about the whale thing. that makes me so sad on your behalf, because when I found out that you had gone and seen that you were out there whale watching, I was actually talking to my husband about it, and I was like, man, we gotta do whale watching at some point, because I'm so jealous that he's out there right now getting to see these whales, and it breaks my heart on your behalf knowing how interested you are in them, that it was not as successful of a trip as you would like. Now, second, obviously, to me, I'm delighted by this. I think that I am so pleased to hear that you've sort of altered your position on this, even if it's not to a fully fledged, I'm like an old Earth creationist now. To me, right, I think that I see this as progress, right? That probably doesn't come as a surprise to you. To me, what this says is that the presentation that I gave you was compelling enough to say, okay, there is support for this, and the glut, let's say, of the evidence seems to support the old Earth over the young Earth. Now, I'm not going to sit here and cast shade on individual young Earth creationists, because frankly, I don't know some of the ones that you mentioned. I've never met Danny Faulkner, and I know young Earth creationists that I actually like a lot and have some respect for. I would recommend Paleologos, Peter, and Todd Wood, and quite a few of the guys associated with the New Creation blog. I think that they do good work, and I like what they do, and I like what they say. Of course, I think they're really wrong on this kind of thing, which sort of comes back around to what you just said there. How is it that we can have respect for someone's intelligence and think that they're these brilliant people and think that they're still so wrong on something? And I think that that's a conversation that I want to have, but I think it's something that we could probably dig in a little bit more, like when we discuss things sort of at the end, because the easy answer is I think they're mistaken because in my experience, and I think I have decent amount of experience talking to young Earth creationists and reading their work. Actually, I take issue with one thing that you said where you were like, we tend to consume evident or content that only agrees with us. I am counting myself out of that because I think I read a lot of young Earth creationists.
Oh, content. I believe you. Oh, yeah.
And because I'm interested in what they say. And now, to be fair, obviously I go in with my perspective already set in place. So it doesn't completely remove me from it, but I do try to consume everything that they put out because I want to know where they're coming from and I want to know what kind of arguments they're making. And that leads me to the last thing that you said. You talked about 3 sort of supports that you see that exist for young Earth creationism, including the faint young sun paradox, coastal erosion, and biological material in dinosaur bones. And I am going to say, just food for thought, like for moving forward, I do take issue with this being presented as support for young Earth creationism, because when I see these, I've looked into each of these separately on my own prior to this conversation. The coastal erosion one I have heard of before. In fact, it's an old enough, what's the word I'm looking for? An old enough sort of contention that it has like a section on the Talk Origins blog, which is this sort of ask boards that existed a while back for the creation evolution conversations in like the early 2000s. So like this isn't a new argument. I have heard it before. And the simple answer to it is erosion rates vary. And also deposition happens. And also the plates themselves are moving. So something can still be there. Like it's sort of like the example that I used last time we spoke, where it's like, we're asking how long it takes for the sand in the top portion of the hourglass to dispel into the bottom portion. And we can estimate that so long as we know that no one's adding sand to the top. Because if someone's adding sand to the top, then it's just going to keep dribbling down into the bottom portion of the container, and there's no way to tell when the hourglass is going to be finished. You need to know the rate, you need to know the starting condition, and you need to know the ending condition to get a good idea of how long something should take. So I take issue with the coastal erosion thing. The faint young sun paradox, I'm not as familiar with, but I do remember looking into this many years ago, and there are sort of hypotheses that explain this from a perspective of nucleosynthesis, I guess, and things of that nature. The biological material one I do take issue with, though, because we have experimentally shown that we can get these biomolecules to last a lot longer than we previously thought. And I think it's such a great example of science kind of acting how it should, because it's like we're presented with something that was so contrary to our expectations that, as I said in the last presentation, Schweitzer was crucified for this. I mean, they took her to the wall for some of the stuff that she was saying initially, but she supported her work well. And so she had enough support at the beginning to basically say, okay, like you can look into this more. And she made her point. And now we find soft tissue a lot. Like we really and truly do. But we have many mechanisms that I think can explain the nature of it. So when I look at these 2 lines of evidence, I wouldn't dream of sitting here and saying that young Earth creationists don't have arguments for a young Earth. But when I look at the counters that I see for old Earth creation, or old Earth evidence, like radiometric dating, like faunal succession, things of this nature, I don't find them nearly as strong as the alternative models that exist for the young Earth creationist arguments that the Earth is young, if that makes sense. Like it's sort of like I see very plausible models that exist for, and I mean this, every objection that I've seen from young Earth creationists, but I cannot say the same for their counters for old Earth creationism. My problem too is that oftentimes, like Danny Faulkner's a really great example, because like he's an astronomer, so he does most of his work in astronomy. And some of the best counters to specific models in young Earth creationism, because young Earth creationism has different models for like where's the flood boundary, or like how does the action during the flood like yield the geologic column and things of that nature. And there are different models that exist that some, maybe AIG supports one and ICR supports another, and they argue amongst themselves as science goes to decide which one has more support for it. And Danny Faulkner has made some of the most incredible takedowns, I guess, of models that he doesn't agree with within young Earth creationism. So I wouldn't dream of sitting here and saying that he doesn't do good work, but I do think that he is, along with Wood, Marab, and Baumgartner and some of these other guys, honest about the portions of young Earth creationism that require sort of a miracle to make sense of what it is that we see. Because at the end of the day, there is evidence that exists that we can sort of judge by our worldviews, and then also by the data itself. And sometimes that data is agnostic, and sometimes it's preclusionary. And what I mean by that is I say it doesn't allow for the possibility of the other perspective. And I would argue that things like radiometric dating, just picking that one as my primary one here, they don't just support the old Earth, they make the young Earth impossible. Right? Like the Fane-Youngstown paradox doesn't make an old Earth impossible. There are like models that exist that do explain it, that have varying levels of support, and we're not sure which one actually makes the most sense. Same thing with erosion, right? Like we know of mechanisms that exist that can redeposit sediment, but we don't know of any way to make sense of the just incredible amount of radiation that is evident in the rocks from a young Earth perspective that isn't going to like vaporize the granitic crust of the Earth 2 dozen times over. So for me, I'm delighted by like where you're at, and I'm so glad that you said. I actually thought that was a great little graphic. I'm glad you took the time to do it, and I appreciate it because I know how annoying PowerPoint can be. But I do think that the more that you look into this stuff, and I'm so glad that you're going to be like, I'm really pleased to hear you're interacting with the Reasons to Believe guys, because I know I said this to you off air, but like I think they're great. I really like all the guys who are at Reasons to Believe. I've had nothing but good interactions with them, and I had the pleasure of meeting Hugh Ross when I went out to California with them a couple of years ago. I'm really looking forward to seeing where you're going to land on all of this. Anyway, awesome, so that's my 2 cents on all that.
I'll say one quick thing. We are going to have another conversation at some point. I don't know when. We can push it all the way to the end about dinosaur soft tissue.
Yeah.
And the reason why is I'm not ready to have that conversation yet. But I will say I do have a friend who is a young Earth creationist, and he just had last year a paper published in a secular journal on dinosaur soft tissue. And according to him, I haven't read it, I don't understand it, it's pretty significant. And so I have to get up to speed on all that stuff, and then we'll have a conversation about it.
I would love nothing more. And, if we, at the end of all of this, like if we want to have occasional get-togethers, like nothing could be more fun to me. And like, I don't doubt that people, I mean, obviously people are finding soft tissue and they're finding it in old material. My challenge is that, can we find soft tissue in something for which there's no mechanism that could exist under an older Earth? You know what I mean? Like, it's like if you found, like, let's say you found like actual wet blood or something like that, like fully complete, like non-altered wet blood. At present, I am aware of no mechanism that could do that from stuff that's 65 million years old. That would be something. But the kind of stuff that we have, at least the stuff that I've seen, and I haven't seen this paper either, so I don't know what's in it. None of it is like totally inexplicable to me. And, they're sort of the, like, I'm sure you would appreciate this because this is just kind of the nature of the beast, but like not all evidence is equal, right? Like not all data points are equal. One odd thing that you find does not immediately overturn everything else. And any alternative model has to be able to explain the rest of that data too. So it's like, that doesn't mean it should be ignored either, though. Like, that's another point. That doesn't mean you get to just dismiss out of hand the oddity and say, oh, it's weird. Like, we don't actually care about that. If we'd done that, we wouldn't know what we know now about soft tissue. So, that kind of stuff, attitude stifles science, I would say. Awesome. Cool. Are you ready to talk about whales?
I am.
Like actually evolution.
Yeah. Whales is one of my favorite topics, so I'm excited.
I know. And actually, so this is kind of interesting. Normally, So normally when I teach deep time to my students, I start ancient and move modern, but I couldn't resist. I was like, I think we're gonna have to start like sooner and move backwards because I want to talk about whales so badly. I think they're so weird, and I have as I said, a brand new appreciation for just how weird they are. And like they're just so such an incredible case study, like such an incredible I'm like missing my words all day today. They're so emblematic of this particular discussion, right? Like this concept of like creation, evolution, or I should say like strict intelligent design versus like evolutionary processes. Whales are really weird. And like evolution has to make sense of them. And this has been something that we've known since Darwin, because you've got these mammals that live in the water. And evolution claims that we come from these Sarcopterygian lobe-finned fish who come from the water, and whales go back. And how did that happen? And why are their bodies so different? Why are they so strange? And of course, they're photogenic, because a lot of the earliest whale fossils that we have, like this basilosaurid that you can see in the picture here, are found in like the Middle East. So you've got these great big whale skeletons just sitting on this sun-baked landscape, their bones cracked and drying in the sun. And there's something about that that's just so poetic. So I had to skip and start with these guys. But I do have some introductory material for this sort of unit, if you will, of our conversations to sort of set the stage for how we can differentiate these hypotheses. And I'm sorry about my dogs, they're barking. So what we're going to be doing today is doing case studies through time, and we're going to start with cetaceans, and I'm going to preface that with some introductory material. A little bit about me. This is me. If you're here, hopefully you know who I am, but I'm a fifth-year PhD candidate in biological anthropology, so I study human evolution. And the purpose of this series is to teach Will Duffy, my guest, or anyone watching, what evolution is and why the scientific community is convinced by it, as well as how it specifically applies to humans. And you guys will probably all notice I have like very, very, very intentionally not mentioned humans or human evolution or human genetics or anything of that nature. And the reason, hold on, they're going nuts over there. They're just so excited by this topic. And the reason is because I'm hoping to save that sort of for the last unit, which is all about humans in particular. Now, this series is like a hybrid of the course that I taught at university. I taught Introduction to Biological Anthropology for 3 years at the lab level, and then for 1 year doing honors lecture. And so we're going to be going over the support for evolution and the ancient age of the earth, now to a lesser degree. This is not meant to be a creationism debunking series. I always feel the need to say that because sometimes people email me and they're like, why didn't you cover this? Why didn't you cover this? And it's like, that's not really the purpose of this. I'm trying to explain why people accept evolution, not, poo-poo all over creationism. But that doesn't mean that we're not going to talk about it where and whenever it's relevant. Here's our roadmap, right? So we're here in March. We're doing our introduction to deep time and cetacean evolution. We have finished Unit 1, which pat ourselves on the back. That's awesome because now the stage is set sort of for the rest of this. So where are we now? Like, I like to start these and sort of orient us in time and then also within this series. I think it's safe to say that given the series so far, we know that populations change over time. And we know that they change over time due to the mechanisms of evolution, right? Mutation, natural selection, gene flow, genetic drift. These are the mechanisms that are behind why a pocket mouse will, over time, go from being tan on the population level to being black on the population level. Mutations give them the variability, and then selection acts and pulls that population in a specific direction. We also know that the amount of time we have for these types of mechanisms to actually take place is evidently immense. We have hundreds of millions to billions of years. And what I'll say here is that at the very least, we know that we've got, call it like 550 million or 600 million years, because that's where we actually have like these really solid fossils found within. Like we've got these living things that are living in these structures that take a really long time to form. And so that's the time that we're going to sort of work with moving forward. Now, these observations set up a ton of predictions, which predictions are, of course, our gold standard of science. And these predictions must be accurate in order for evolutionary theory to be true, at least in the sense that we say any theory in science is true. And I'm going to summarize it with this slide here. If evolution over billions of years from a last universal common ancestor explains modern biodiversity, so all of the different critters that we have alive today, many fields must independently come to the same conclusion. So what do I mean when I say that? If all tetrapods or land-dwelling critters that have 4 legs, sort of developmentally speaking, so that's going to be reptiles and amphibians and mammals and birds, if all of us descend from these lobe-finned or sarcopterygian fish, then a lot of stuff has to be just obvious and evident within our sort of scientific literature. Now, we've already shown the first two. We've shown that we have these mechanisms that can explain the emergence of new variations or traits, and then we know why those traits do or do not proliferate. And we also know that from geology and physics, as I said previously, we seem to have a lot of time. But that's not going to do it, right? Because showing even if the changes themselves are quite large in the present, we have like millions and millions of those large-scale changes that need to be demonstrated through various different mechanisms to support evolutionary theory. The first is through taxonomy. So taxonomy, which we'll talk about here in a second, is sort of how we categorize life. And all life, from a taxonomic standpoint, to support evolution, must form a nested hierarchy in functional and non-functional regions of the genome alike. That's a lot. We're going to dig into that and dissect it a little bit more here in the next couple of slides. We also must show that development and morphology, so the way that critters develop in utero or in the egg and their physical characteristics now, should support the evolutionary relationships that we're proposing, right? So if birds descend from dinosaurs, there should, or I should say from non-avian dinosaurs, there should be some traces of that dinosaurian past in modern birds. From paleontology, we must see, for evolution to be, true, the emergence of the characteristics that define modern forms show up in the fossil record over geologic time. And via genetics, we need to be able to pinpoint the genetic changes and the pathways for those changes for the evolution of key traits via theory and experiment. So like, let's say we were to find Tiktaalik, as this little cartoon here at the bottom depicts. We need to be able to show not just that that fossil exists and seems to support this transition, but we need to be able to go to genetics today and show like genetically what needed to have happened for that change to actually occur. Which mutations did we need? How did it impact development? And so on and so forth. So to put this a lot more simplistically, for evolution to be well supported, we need to have the mechanisms of evolution and the time for evolution, which, as I said, I'm sort of giving us a check mark for Unit 1 here. We need to see the pattern of evolution. We need to see the traces of evolution, and we need to have the pathway for evolution. So living things need to form these sort of family trees. We need to see the traces of past relationships in living organisms, and we need to show how that could have actually happened step by step genetically. So for example, in this image over here, you can see a cat with polydactyly. So some cats will have extra little digits, and instead of having 5 digits, they'll have like 6 or 7 on each paw. And we've managed to find out which genes create that phenotype and how that phenotype comes about. And I think that needs to be well supported to propose some of these major changes. And I think that's fair. Tell me if you think that's not fair as we sort of move forward. All right, so taking a step back, systematics, and this is going to be sort of our taxonomy, I should say, breakdown, is the study of naming and classifying life. The purpose of classification in general is to convey information. The reason we put things in boxes is so that we can communicate what we're actually talking about here. The modern version of systematics and of taxonomy was started by Linnaeus in 1735. You should probably remember him from a couple of lectures back. And he's the guy that devised this system: kingdom, phylum, class, order, family, genus, species, or the basic taxonomic ranks by which we organize living things. Organisms occupy increasingly specific categories as we move from these higher taxonomic ranks down, and they get broader as we move back up. This is exactly analogous to Russian nesting dolls, right? So you've got like the littlest one that occupies all of the larger nesting dolls, and as you open them up or close them up, you're moving up and down that nesting scheme, which is why we call it a nested hierarchy. A lot of times this concept can be kind of difficult to wrap your head around. And so I'm going to pitch it to you sort of as I pitch it to my students. We've got a bunch of different critters here, a bunch of different organisms. We have a paramecium, a snail, a snake, a fish, a baboon, a skunk, a chimp, and a human. And then over to the far right, we have our categories within the sort of Linnaean taxonomic system. Here's how we devise this. All of the organisms in the blue box that you can see right now belong to the domain, as indicated over here, Eukarya. And the reason they belong to that domain is because they have the characteristics that define eukaryotes. All of the guys in this box have cells that have a true nucleus, which are bound by a membrane, specialized organelles, etc. Getting a little bit more specific, all of the organisms boxed in this light blue color belong to the kingdom Animalia. So you'll notice the paramecium has been kicked out. And the reason is because the characteristics that define the kingdom Animalia include being multicellular, having a blastocyst as a part of your development, being heterotrophic, and many other characteristics. And the paramecium doesn't have all of these traits. So it doesn't belong to the kingdom Animalia. That doesn't mean it's not still a eukaryote, but it does mean that it's not an animal. Moving more specifically still, all of the boxed organisms in this light green color belong to the phylum Chordata. And now the snail has been sort of booted from this club. And the reason is because to be considered a chordate, you need to have a notochord, you need to have a hollow nerve cord, you have to have a postanal tail, you have to have pharyngeal slits, and you have to have a thyroid or an endostyle. And you can pick up a snail or an octopus or a spider for that matter, and you'll know that none of these guys are chordates because they don't have any of those characteristics. That doesn't mean they're not mammals, and that doesn't mean they're not eukaryotes, but this is where the chordates split from everybody else. Next, we move down to the class level. This is class Mammalia. Now, snakes and fish have been booted from the club. Mammals produce produce milk. They have hair, they have 3 inner ear bones, and they have a dentary squamosal joint, amongst other characteristics. These are the traits that define mammals. You don't find all of these things in snakes. You don't find all of these things in fish. And yes, I know what people in the comments are saying about humans being fish. That's another story. Let's call it a clownfish, for example. Next, we move more specifically again to the order primates. Baboons, humans, and chimps are all primates because we have dexterous hands, binocular color vision, usually big brains, a post-orbital bar, and we have nails on our fingers instead of claws. And specifically, that's on our thumbs and on our big toes. So skunks are still mammals, but at this point, they get booted off into their own category. They're not primates. More specifically still, we have the family Hominidae. And as a pro tip, anytime it ends in DAE, it's a family level. That's like the root for family classification. And the reason why baboons get booted here is because unlike humans and chimpanzees, baboons don't have the huge brains, the slow growth system, the broad chest, the long arms or the Y5 molars, and of course they have a tail. Humans and chimpanzees have all of these characteristics. Baboons don't, so they've been kicked out of the club. Finally, humans belong to the species Homo sapiens. Homo sapiens is the only species alive today that is bipedal, that has an enormous brain, that has complex tools, that grows up super duper slowly, has little canine teeth, and so on and so forth. So you're seeing, like, as we've gone from really, really broad to really, really specific, more and more animals have been kicked out until we arrive at the species level, or the most specific of the taxonomic rankings. But we can roll this back. So humans, all humans alive today belong to the species Homo sapiens. For sure, there is no doubt every human that is living today is a member of Homo sapiens. All Homo sapiens belong to the family Hominidae. All hominids belong to the order primates. All primates belong to the class Mammalia. All mammals belong to the phylum Chordata. All chordates belong to the kingdom Animalia, and all animals belong to the domain Eukarya. What this means is that if you find something and you know that it's a mammal, right, you know that in addition to being a mammal, it is definitely a chordate, an animal, and a eukaryote. But anything more specific than that level, you're going to have to investigate further. This is the nested hierarchy, and this is how all life, as we can tell, is organized. So what you can see on the screen now is an ostrich, my dog Teddy, and then me, and our classification at the kingdom, phylum, class, order, family, genus, species levels. And when we look at all of the characteristics that these 3 organisms actually have, what you'll find is that the 2 that are the most similar to one another are me and dogs. Humans and dogs are both mammals, so they share more in common when you're counting up all the characteristics an organism has than either of us share with an ostrich. This is the sort of nesting pattern that you see, and it forms this little family tree there at the bottom. When all of the characteristics of living things are considered, they form nested hierarchies. So like, it's critical to put this into context. We're talking about suites of characteristics, not individual characteristics. So take like a pangolin, for example. Pangolins have scales, and like no other mammal has the type of scale coverage that we see in pangolins. We usually associate scales with something like a reptile. So does that mean that pangolins aren't mammals? No, because pangolins have all of the characteristics that define mammals. When you look at all of the traits that they have, they nest with the mammals overall, even if they have scales as a reptile would. And, that's sort of simplifying it, because the types of scales that they have aren't the same types of scales that you see in reptiles anyways. But the point of that is to sort of demonstrate that the bulk of the characteristics is what matters. And going from broad to specific is how you end up with these sort of addresses for each individual organism and each species. This is exactly the same type of thing that we see with languages. So I know you like the language analogy, so I decided to use it again. Here we have a family tree for a bunch of different Latin languages. So let's just start with French. We have French here at the bottom, outlined in this little box. French is a type of Gallo-Romance language. It's also a Western Romance language. In fact, all Gallo-Romance languages are in fact Western Romance languages. And all Western Romance languages are Italo-Western languages. And all Italo-Western languages are Continental Romance languages. And all Continental Romance languages are Vulgar Latin languages. And all Vulgar Latin languages are Latin languages. This is a nested hierarchy. Simplistically, you can create nested hierarchies with a lot of different things. So what you can see on your screen right now is we have nested hierarchy formed with vehicles. So we have different types of vehicles nesting into the water, land, and air category. And if we zoom in on land, it might have one wheel, which would mean it's a unicycle, or two wheels, which means it could be a bike or a motorcycle, or four wheels. And there are a lot of possibilities here. You could say that's a nested hierarchy, but the problem is it's not like a true nested hierarchy, because true nested hierarchies are linked by descent with modification, and that modification part is critical. In both living things, as seen in all of these dog breeds here, and in languages, each individual species or language is connected through time by tweaks in the genome or in the vocab or grammar. You don't see that with design nested hierarchies, as we saw up here with these vehicles. There's no through line there. You can count up the characteristics that they have, but it's not like what we see with genetics or like what we see with languages, where you can actually use sort of the track changes function over time and sort of trace the history. So we can ask a question next then, right? Like, I've created these nested hierarchies. What on earth makes us think that the nested hierarchies that are formed here are indicative of relatedness, right? Like, just because wolves and dogs share a lot with each other doesn't mean dogs descend from wolves, not on its own, right? We have to show more than just that. Under evolutionary theory, nested hierarchies reflect levels of relatedness. And this is something that Darwin predicted all the way back with On the Origin of Species in 1859. He predicted that all life could be traced back to one or a few universal common ancestors, with all of modern biodiversity being the result of descent with modification. So critters will experience mutations, he didn't know that at the time, and then selection will select specific variants which are more successful. They'll pass those traits on, and the population will change over time. Things descend and are modified. The thing is, everybody agrees with this. Everybody agrees with this concept to a degree, because this is how family trees work. So you can see in this family tree for Napoleon, you have the exact same pattern as a family tree for species, like on the species scale. But family trees are devised by actually observing human descent with modification and then documenting it via historical records. You can watch somebody give birth and you can say, she gave birth to this guy, this is her son, and you can do that ad nauseum, and people have always been around to observe these kinds of things and record it. So what then allows us to do that with species? Why is this sort of an analogous situation? Without question, genetics reflects relatedness in organisms, at least insofar as we can tell. The reason we know this is not because we assume it. The reason we know this is because we've tested this directly. we've taken real known family trees for organisms, like in this experimental paper here. I believe we did it with specific types of bacteria, but you can do this with human families or dog pedigrees or whatever today, and we have done this. Real known family trees, and then sequence the genomes of the individual specimens who are within that family tree, and they match. The family tree that we know via observation matches the family tree that we create via genetic similarity. And this relationship is like so non-controversial. It's such a given in biology that it holds up in court. It helps us catch criminals. It helps us save lives medically, and it reconnects families. These databases, like from ancestry.com, for example, are just enormous. And what I'll say is that ancestry.com is kind of unique and cool in this way, in that it doesn't just take your DNA and like do a family tree based off of who else has submitted their DNA and sort of give you your results that way. They cross-check it with historical records. And the reason they can do this is because those things typically match, unless someone is being dishonest about who their father is, for example. We can do this at the population level too, though. You don't just have to do it with individuals within specific populations. You can take representatives of populations and determine how close groups are as well. These similarities can then be sort of combined with each other to give us like these molecular clocks to know when specific characteristics or mutations emerged in which population. And then we can take that back in time using known mutation rates to find out when groups split off from one another. And we tested this with the examples that you can see on your screen right now. So we can look at the population structure in post-colonial North America. We can look at when the indigenous people of the Canary Islands arrived and how their populations have changed over time. We can look at how the migrations in the Iberian Peninsula sort of map with our expectations of historical, or our expectations, I should say, from historical records. And we can just trace human migration as a whole. These population-level family trees are called phylogenetic trees. So as you can see, this is sort of a family tree tilted on its side. And instead of individual people or individual dogs or individual bacteria, we're looking at people groups. And what's really, really cool about this, and the reason I've included some of these papers in the back, is because these are checked, again, with historical records and correspondence. So this is another sort of line of support that these phylogenetic trees are reflective of reality as we've observed and recorded it. And of course, it doesn't just work in humans. Phylogenetic trees work on all other species, like without exception that I am aware of, and they're commonly used in the agriculture and companion animal industries. So I've pulled here a paragraph from the AKC's, the American Kennel Club's DNA profile program, as well as this pedigree here listed up top between the sire and dam of a specific German Shepherd. That might be a Belgian Malinois. Yeah, it's a Belgian Malinois puppy. And this is also how we know which dog breeds are more similar to which other dog breeds and where they were bred, like first sort of invented, bred into existence, and what they're most similar to. Phylogenetic trees of living organisms, like the ones for people groups or the ones for dog breeds, assume only that genetic similarity reflects relatedness. So, for example, without testing our genomes, I think it's probably a pretty safe bet that my genome and my sister's genome are both more similar to one another than either of our genomes are to our cousin. But the three of us are all more similar to one another than any of the three of us is to a stranger. And a really critical part of this is that this isn't testing part of the genome. This is the whole thing. You can't test more than all of it. And when you sequence the entire genome and test them, we know, at least without exception that I am aware of, that the more similar two genomes are, the more closely related two organisms are.
Okay, question.
Please.
Is this a straight percentage we're talking, or is it something else? Because I thought humans were all 99% similar.
We are. But in that, like your genome is billions of base pairs long. So 1%, even in the protein coding region, is a lot of room for variation. So that variation within the percentage in which we do differ is how these trees are constructed. It's kind of like if you have two Word documents and they're 100 pages long, but the last page is different in these two documents. You can get a percentage for how similar they are by just comparing those last two pages. Does that make sense?
Yeah. Okay. So again, I want to make sure I understand this. So
Please.
if two people are 98% similar and two people are 97% similar, the 98 are closer related, more closely related. Is that I mean, I know I'm dumbing it down, but is that what we're saying?
In like super simplistic terms, yes. The more you share genetically, the more closely related you are. So if you have a brother, right? Or like let's just go with the brother. Like let's say you have a brother and you sequence the genome of you, your brother, and 1 million other people on the planet randomly selected. There is a 100% chance that you will share the highest percentage of your genome with your brother. And the reason is because we inherit DNA from our parents. So you and your brother both inherited DNA from the same parent. So you're going to share the overwhelming majority of your DNA. The bits that differ are going to come from like recombination and mutation only. Whereas the split off point between you and various other strangers is way deeper in the past. There's a lot more time for mutations to make them more different. Does that make sense?
Mm-hmm.
Okay.
Yeah. The next line, which you haven't read yet, to my knowledge, this has never. Sorry, one more back.
Yeah. Sorry, please.
To my knowledge, this has never been observably violated in animals. What does that mean?
So what I'm saying here is that I believe that it's possible that there's some kind of like animal out there that's multicellular, but not like that's small and non-complex, like something similar to an amoeba, but multicellular. And maybe they have the ability to do like what bacteria do, which is horizontal gene transfer. And then that would mean that they could exchange DNA without actually having to sexually reproduce, which would kind of ruin this simplistic explanation of genomic similarity, because you can just exchange DNA while you're still living. And so I don't think anything like that exists. But the reason I've said this is because I'm always finding exceptions to things that I find in biology. But to my knowledge, there is not a case in any animal where an individual and their sibling are less genomically similar when you sequence the entire genome, billions and billions of base pairs, that they share less than one of them does with a stranger. I am aware of none of that. I mean, that would be pretty groundbreaking.
Okay, and is the prediction being made here, which has already been tested to some extent, that as we sequence more genomes, it will confirm the nested hierarchies that were created based off phylogeny?
These are the phylogenies. So the family trees that we create from an evolutionary perspective can be done by shared physical characteristics, right? So why are humans considered apes? Because we have all of the physical characteristics that are used to define apes, right? But at the same time, there are many ways that humans differ from other apes. So we could ask the question, okay, like when you look, and I'll be talking about this later on when we do like human evolution, but if humans, just bear with me here, get on board with the assumption, and let's say that humans are in fact apes. Then there's humans, and there's chimps, and there's gorillas, and there's orangutans, and there's bonobos, or a bunch of other types. At face value, we should look at the group of us, and we should say, okay, out of all of those guys, humans are probably the odd man out because we look the least similar to everyone else. But when you sequence our genome, orangutans are the odd man out. Humans and chimpanzees share more of our genome than either of us does with an orangutan, which is weird. It doesn't make a lot of sense. Because normally you think of physical characteristics reflecting one-to-one of the genome, but they don't actually do that. It doesn't actually function exactly like that. But what I'm trying to sort of demonstrate here is that the family trees that we create, like throughout all of history, from medieval times to now, from Roman times to now, from the first civilization, the family trees, the pedigrees that we create by observing who gives birth to who, that is a one-to-one match with the family trees that we create by sequencing genomes. And so what that means is that genomes in isolation can tell us how similar two individual people are, or how similar two individual dogs are, and thus how closely related they are, because this is, insofar as we can tell, a one-to-one relationship. And so as we step back, as I'm going to sort of show you here in a second, we can do this with species too, and we don't know where that stops or why it would stop. I think that's the challenge that creationists face. This is something that you might remember from my conversation with Fuzz when we were talking about Neanderthals, right? Like, I believe I think that Neanderthals are a separate species from humans, but we could still interbreed. So does that make sense? Can I move on to the next slide and maybe it'll make a little bit more sense?
Yeah, definitely. I like this because I think this is an area where I think creationists should be willing to make a prediction that they think evolution will be wrong, and then we'll see.
I think so too. And we'll
Yeah, I like
see where that goes. So here's the question, right? If genetic similarity, insofar as we can tell, like just full-on genomes, the person that your genome is going to be most similar to is going to be a family member instead of a stranger. And then you and your family members and your extended family are all going to be more similar than any of you is to a stranger, and so on and so forth. If genetic similarity equals relatedness, where does that relationship actually stop? So here we have a couple of dog breeds. Like, I don't think it's controversial for me to say that golden retrievers and German shepherds and chihuahuas are all dogs. And so they're all related. Like, they're actually related. They share a common ancestor. And we can sequence their genomes, and we know that they do. In fact, we can find out when specific breeds actually emerged and where they likely emerged by sequencing their genomes and calculating, using the molecular clock, when specific genes that characterize these breeds show up. So let's say with these cartoon dogs, we sequence their genome and they form this little family tree where the golden retriever and the chihuahua are more similar than the German shepherd over here, or the wolf dog, or whatever we want to call it. Dogs, all our dog breeds, are most similar. Like, if we want to say, what's the animal that all dog breeds are most similar to, it's going to hash out to be a wolf. And this makes sense because dogs were domesticated from wolves. So all dogs are most similar to wolves, and they're more similar to wolves than they are to anything else. Dogs and wolves are more similar to each other than either is to a coyote. So you can sequence the genome of every dog breed on the planet, and also wolves, and those guys are all going to share more with each other than any of them does with a coyote. Likewise, dogs, wolves, and coyotes all share more with each other than any of them does to a fox. Right? And that makes sense, right? This tracks with what we know of these animals, and it's reflected in their genomes too. And finally, all canids, or members of the dog family, are more similar to one another than any of them is to a cat. So we can ask this question, right? Like, I should say too, all carnivorans are more similar to each other than any of them is, for example, to a human. So a cat is really different from a dog, but genetically they share more with a dog than they do to a human. And not just genetically, physically, like their physical characteristics work the same way. So where does this stop? Right? Like, where is the line drawn with genetic similarity? From a creationist perspective, where would they say, okay, I'll accept that genetics unites all humans, and I'll accept that genetics unites all dogs, and that it unites dogs with wolves, and maybe they'll even say it unites dogs, wolves, and coyotes, but where does it stop working? Now, from an evolutionary perspective, we make really plain predictions here. If dogs and cats are actually related to one another, then taxonomically speaking, they should form that same nested hierarchy that I've been talking about all this time, like you see on your screen right now. And that nested hierarchy should exist in the functional and non-functional regions of the genome alike, in the parts of the genome that do stuff and in the parts that don't. And I have an analogy that I think will maybe help with this here in a second. Likewise, if dogs and cats are related and they all belong to an order of animals called carnivora, carnivora is an order just like primates is an order, it's sort of a broad category of mammals, then dogs and cats, and in fact all carnivorans, should share features of development, their physical characteristics, morphology, and genetics should all demonstrate together that this evolutionary relationship is feasible. There shouldn't be, amongst all of these guys, if they're actually related, some kind of mutation or some kind of physical characteristic that we can't figure out how that could possibly have come about. Now, all living carnivorans, or members of this sort of carnivoran group, are characterized by a type of tooth called a carnassial. So in this little picture down here, this is what carnassials are. If you have a dog or if you have a cat, you can open up their little mouth and you'll see these carnassial teeth. No other living group of animals has carnassials outside of the carnivorans, which means that if you find carnassials, it's a carnivoran. That's the characteristic that we use to define them. And this has been the case since before evolution was even proposed by Darwin, all the way back with Linnaeus. So from a paleontological perspective, in addition to all of these other characteristics, we should see the emergence of the characteristics that define these modern groups, like dogs and cats, emerge in the fossil record over geologic time. What that means is that if they have a common ancestor, from an evolutionary perspective, that common ancestor probably isn't a dog and it probably isn't a cat. Instead, it's something that has some features of both and develops over time as these things, as you move through geologic time. We should see the emergence of the traits that define them from an evolutionary perspective. Now, I feel like we need to talk about transitional species before we move any further. So, what is a transitional species? Definitionally, transitional species are usually species that link proposed ancestor and descendant groups morphologically and chronologically. So they link them together in space and time. Here's an example that I made up. Let's say we have these two mammals. I made these goofballs up. The one on the top is this little guy called Silitherium caudata, which means little beast with a tail. And the one on the bottom, the bigger, scarier looking one, we'll call him Gubertherium maximus, which means like the big Gubertherium, the big goober beast. Let's say that in the fossil record, we find Silitherium caudata from 5 million years ago. We find the fossils and we radiometrically date the layers that these fossils are found in, and they're 5 million years old. And Silitherium can be defined, like if we were to describe it, it's different from all other mammals in that it has 4 digits on each foot and a unique swinging, bludgeoning tail. So it's got this tail with sort of a knob on it that can use to knock out predators. We find Gubertherium only 1 million years old, ago. This guy's a lot younger, like geologically speaking. And instead of being small, Gubertherium is a big mammal with these 2 digits on each foot instead of 4, as well as big tusks and a single massive head horn for competing, as well as a short but uniquely tough tail. Let's say, from an evolutionary perspective, we look at these guys, since we know time has passed from Silitherium to Gubertherium, and we want to say that these two guys are actually related through time. We could propose, right, that there might be a species that links them together physically and in time. Let's say we find this guy, Tridactyltherium, which means 3-fingered beast, platycephalus, which means flat skull. And we find this fossil at 3 million years ago, in between 5 and 1 million years ago. So it's in the right spot chronologically. And this guy's defined by 3 digits, not 4, not 2, but 3, directly in the middle. And it has a thick cranial pad. It's not quite the horn that we see in Gubertherium, but it's definitely, there's something there that wasn't there in Silitherium above it. And just like the species that came before and the species that came later, it has a little knob at the end of its tail. It's this uniquely thick knob at the end of this tail. These animals are all mammals. We know they're mammals because they have that dentary squamosal joint. They have the specific types of teeth. Maybe we find some really exceptional fossils with preserved hair, or maybe we find indicate, like maybe we get some DNA and we sequence them and we know that they can produce milk. They're all mammals and they seem like they're united through time by this uniquely tough tail tip. That is an example of a transitional species because what it's done here is it's linked the former and the latter in time and in morphology. It seems sort of in the middle. We could support this with additional information though. What if we found out that Gubertherium, our big guy down here at the bottom, when it's developing in the womb, what if for a little bit it has 4 toes? Like what if when the baby starts to develop, the extra 2 toes on each foot start to develop and then they abort? And the toes sort of quit developing and that's why it ends up with those 2. That would probably be additional support that it's related to these 4-toed guys who existed beforehand. Likewise, what if we found out that our little guy up here at the top, Silitherium, who doesn't seem to have tusks, let's say we found a bunch more fossils and these fossils are even better preserved and in the mouth we actually find tiny tusk roots, which seem to suggest that while the tusks weren't very big, maybe they didn't protrude from the mouth, it does seem to have a basic version of what we see 4 million years later. And what if we have more than one species of this guy, Tridactyltherium in the middle? And what if, while most of them tend to have these 3 digits on each foot, what if we found out that ones that are 4 million years old had 3 digits on the front foot, like regular members of its species? But what if on the back feet it looks more like Silitherium? All of that would lend more support to the evolutionary relationship here, because what we're seeing is a morphologic through line. Physical characteristics emerge and then they're retained through time, identifying these guys as being relatives because no other animal has those characteristics. What you could do is you could create a cladogram just like this. And we could say, okay, like let's look at the sort of taxonomy of Gubertherium maximus here. We know that Gubertherium is a mammal, as I said earlier. So because it's a mammal, we also know that it must be a chordate, it must be an animal, it must be a eukaryote, and it must be alive. It must be a living thing. And this bears true for every living thing now. If it's a mammal, it is definitionally all these other things. If it's a chordate, it's definitionally everything above it. This is what we mean when we say nested hierarchy. But what we can do here is we can sort of create this evolutionary relationship, or rather plot it. And we can say, okay, at the base of this little tree that we've drawn here, Silicaudata, or the order, shows up for the first time. And this order is defined by a uniquely toughened tail tip. And the reason that we know that all three of these guys belong to this order is because all three of them have that uniquely toughened tail tip. Likewise, Gubertheriidae ends in DAE, so we know it's a family. These guys are defined by 3 or fewer digits, as well as a toughened cranial roof. Well, this shows up here before the emergence of Tridactyltherium and Gubertherium, but after Silitherium has split off. Because Silitherium doesn't have 3 digits, it has 4, and it doesn't have a tough cranial roof, which means that characteristic evolved after Silitherium split off from the rest of the group. Finally, Gubertherium has the 2 digits, has that big cranial horn on the top of his head, and has these prominent tusks. Those characteristics don't exist in Tridactyltherium, so we know they emerged or evolved after Tridactyltherium split off. And that's how you create this sort of cladogram in this case. This can be applied to a phylogenetic tree too. It's just the accumulation of these characteristics, the morphologic change over geologic time. It's important to note that morphology, the physical characteristics, are probably more important than the fossil's placement in time. And the reason I say this is because we know ancestors don't necessarily go extinct when a new species splits off. So modern dogs descend from wolves, but wolves are also still here. They didn't have to be. If we'd been successful in hunting wolves to extinction in various places through time, it may be the case that what if we only had coyotes today? Then dogs could descend from wolves and wolves are also gone, but in other cases, wolves can persist. The same logic applies to family trees. You don't, like, your grandpa doesn't die because you are born. You know what I mean? Or your great-grandpa doesn't die because you were born. You can outlive the split. So if we go back to this little cladogram here, it's possible that these 3 fossils could be found at the same time. And the evolutionary relationship could still hold as long as Silitherium here has the first emergence. This thing needs to have the oldest of all 3 fossils, and then Tridactyltherium, and then Gubertherium, for it to be like a really good transitional species. Okay, now, a modern analog here that you could draw, and that I've seen creationists draw before, is dog breeds. Because like dog breeds are just incredibly diverse. You could take all of the dog breeds that we have, and theoretically, you could order them in an evolutionary sequence, right? You could say, look, you can see the face gets more flat as you move from the French Bulldog through the Chihuahua to the Pug. Maybe the size alters a little bit, but the face is getting flatter, the palate more compressed. But the problem with that is all dog breeds in this little evolutionary sequence here are alive right now. And all of these dog breeds have the same features. They all have the same teeth. They all have the same bones. The shapes and sizes of those bones are different, but it's not the same as what we saw with our little mammal example here, where we're actually seeing new characteristics being gained through time to distinguish these species from one another. In the case of dogs, nothing new is really being gained feature-wise. We're altering what is existing there, which is what tells us these guys all belong to the same species. Thus, this sequence does not show like a real evolutionary transition. However, it does still show us that these guys are definitely related to one another. They do have all the same physical characteristics. And there's no animal on the planet that isn't a dog that has every trait that defines a dog. So you can kind of think of it this way. Morphology is sort of the proof, quote unquote, that physical intermediates can exist. And chronology, or the location in time, is kind of the proof that these features changed over time, that these features actually evolved. And I've included this picture of pugs here for a reason, because even though we know French bulldogs didn't evolve into Chihuahuas and didn't evolve from pugs, we know that pugs have changed a lot over time. We have these paintings of pugs from when the breed was first sort of invented, for lack of a better term, hundreds of years ago, and they look very different from the pugs that we have today. The tail is less tightly coiled, which you could actually see looking at the skeleton, the caudal vertebrae there. The face is less fat. Well, it is also less fat, but it's less flat. The teeth are going to be less compressed in there. The palate is going to be less orthognathic as well. The chest is less barrel-shaped. So what you could do, because these two dogs did exist at different times, is you could take the skull of every single pug from the moment the breed was invented to now, and you could watch actual evolutionary change over time, because you've got the morphologic change and the geologic time, in this case, the historical record. So taking a step back, are dogs and cats related? There's two hypotheses that we have here. We have common descent, which would state that they are related, and I've sort of set up the predictions for that. And then there's strict common design. And I've placed strict common design, I've sort of phrased it that way because there are theistic evolutionists who would propose that common descent is common design, right? Like you can have common descent that's designed by God or set forward by God. So when I say strict common design, I'm meaning that there's actually a discontinuity between these groups. They did not share a common ancestor. Before we move forward, we have to ask the question, is it even possible that we could rule common design out? So I've drawn for you a little family tree here, a little phylogenetic tree. From common ancestry's perspective or common descent's perspective, the whole thing is going to be a real, actual, reflected relationship. Whether you're using physical similarity or genetic similarity, similarity reflects relatedness, and it always converges back on a last universal common ancestor until you reach the very first one. From the separate ancestry perspective, as outlined by many creationist organizations today, they would propose that what you see in green is real, that those relationships are real, but what's in pink isn't, right? The pink it's an illusion, so to speak. And there's a problem here because the same methods that we use to determine the relationships between these terminal branches to each other, like in this far left sort of subsetted family tree here, a creationist and an evolutionist alike would accept that these three terminal nodes are actually related. They did share a common ancestor. In the case of evolutionary theory, we would say in the common ancestor, they share a common ancestor with this adjacent group, and then these two groups share a common ancestor with each other all the way back to the beginning. Whereas a creationist would say these guys do share a real common ancestor, but at some point, creation happens. At some point, they no longer have a common ancestor. This relationship stops. And the problem with this is we don't see that signal when we look at these guys genetically or morphologically. There doesn't seem to be a point where genetics says, okay, there's some kind of genetic stopgap here that says this cannot be crossed, or like these things are absolutely too disparate to be related to one another. But the problem is somebody from a creationist perspective could look at this and say, well, yes, it does appear to form this real family tree, but that's because of common design. That's because these guys are sharing these physical characteristics, but the physical characteristics are reflected in the genomes. So they're going to share a lot of genetic information, even if they aren't actually related to one another. However, there is a way to distinguish between the two of these. Strict common design can be ruled out if organisms form these nested hierarchies in the region of the genome that are functional and in the non-functional regions of the genome. Now, we can have a conversation about how much of the genome is functional or how much of the genome is non-functional, but insofar as I can tell, everybody agrees that some portion of the genome is non-functional. Whether it's 1% or 5% or 80%, there is a portion of the genome that doesn't do anything. At least I don't think I've encountered creationists who would say for sure 100% of the genome is functional right now. And the reason they usually point to is they'll say genetic entropy or the fall or something like that allows for the accumulation of these non-functional regions. But the thing is, if you have a non-functional, or excuse me, if you have a nested hierarchy, like a family tree that forms when you sequence the functional regions, that makes sense from a design standpoint because they're doing the similar things. So we would expect God to design them in similar ways, and that would be reflected in the genome. The non-functional regions aren't doing anything. So why would the signal be there if it's not reflecting anything that's actually being done? An analogy works a little something like this. Suppose you're an editor for like a major publishing house, and you receive a new manuscript that comes down onto your desk, and it looks really, really similar. Let's say for like a thriller, like a new thriller novel, and it looks really similar to one that you got a couple of years ago, but the author has a different name now. Right? You look at it and you're like, this is really, really familiar. This manuscript reminds me of one that I got 5 years ago, but that one was written by Holmes, and this one's written by Bobby. The story itself between these two manuscripts might be really, really similar, but you notice as you read through these manuscripts that there are major changes between the two of them. Sometimes entire chapters are different between these two individual manuscripts. Now, it's possible that this new manuscript that you've gotten was written by a totally new author. Just, people design similar stories all the time. Maybe they designed a story that was really, really similar to the author that wrote that first manuscript. But then as you read through these manuscripts, you notice that they don't just share paragraphs and chapters, they also share the same typos. And sometimes the typos they share are very, very unusual ones. It seems reasonable to propose that two individuals could come to the same general story and maybe even have very similar chapters, but it seems very unlikely that they would also misspell all of the exact same words in the exact same place down to the page number. That makes you think that the manuscript that you have now, the new one, is almost certainly a revised version of that one from years ago. And the reason is because the flaws carried through from that first manuscript to the one that you have on your desk now. This is kind of how non-functional DNA works. It's DNA that is accumulating, right? It's not subject to selection. So it hangs around in the genome, not doing very much. And if design is reflecting, or if the genomes are reflecting common design, that shouldn't also form a nested hierarchy because it's not reflective of any design. It's just hanging on. So what do we see? Carnivorans, all carnivorans, so all of the cats, including cheetahs and leopards and civets and hyenas, all of those guys form a nested hierarchy within themselves, the family Felidae, and that family forms a nested hierarchy with Canidae, the dog kinds, the bears, the seals, the walruses, the skunks, the raccoons, the muscelids, like the mongooses, all sorts of stuff like that. These guys form a nested hierarchy together, a mutual family tree. And they form this family tree in the regions of their genome that are actively doing things and in the regions of the genome that aren't. Likewise, the common ancestry of all carnivorans, which again is this entire group of critters here, the guys who have those carnassial teeth, isn't that difficult of a transition to actually propose. So I've included here a bunch of different members of Caniformes, or the guys on the red side of this phylogenetic tree here, as well as members of Feliformes, or the blue guys on this phylogenetic tree here. Caniformes means dog-like, Feliformes means cat-like. And generally speaking, caniforms are defined by having like longer snouts, more teeth, like your dog is going to have more teeth than your cat does. And also, dogs don't have retractable claws. Virtually all of the feliforms have retractable claws, which is something that shows up in their skeletons. And there's minute aspects of their auditory bulla, or like their sort of hearing complex. So these two are definitely different from one another. But when you look at the skeletons side by side, this just isn't that big of a jump, right? Like it doesn't seem very difficult to imagine, if you will, that a mutation could allow them to gain teeth, or a mutation could allow them to retract their claws, or a mutation could shorten the snout, and so on and so forth. This just isn't a major difference. There's not a lot of ground to cover here, especially in the context of some of the mutations that we discussed earlier in this series, where single mutations Large-scale deletions or large-scale duplications can drastically impact, in the case of some of our arthropods, the number of limbs or the number of wings that they have. This isn't a hard sell, is what I'm trying to say. It's feasible evolutionarily speaking. Paleontologically speaking, carnivorans show up slowly. So according to paleontology and according to evolutionary theory, if dogs and cats share a common ancestor, they should show up slowly through geologic time. The very first carnivoran that we see is called a myacid. Members of the group Myasidae, I think it's Myasidae. These guys are primitive carnivores, and we know they're members of the same carnivoran group as dogs and cats because they have those shearing carnassial teeth. The big thing that defines carnivorans, these guys have it. And that is the only thing that sets them apart as actual members of carnivorans. They don't have any of the additional features that define dogs, and they don't have any of the additional features that define cats. They don't have specializations of the auditory bulla, they don't have retractable claws, and they have more teeth, if memory serves, than both dogs and cats. But the very first dogs and the very first cats, so the very first skeletons that we can find that belong to Feliformes or Caniformes here, because they have those characterizations of the face, teeth, and auditory bulla, they're like barely distinguishable. So you have just a scant couple of carnivoran characteristics emerge in Myacids, and then probably about 10-ish, 20-ish million years after that, we see the emergence of the characteristics that actually define not just carnivorans generally, but specifically members of the dog group and the cat group, which is really cool. So that's compelling-ish, but that's also like just jetting through a couple of characteristics. And it's also for a relationship that's not very hard, right? Like dogs and cats actually being related to one another. They both have 4 legs. They both have 4 types of teeth. They both have tails. Like they're both meat eaters. They're just not that different from one another. We need a lot more to actually support common ancestry of all life. So what I'm going to do to show support for that is over the next 3 lectures, the rest of this one and then the next 2, we're going to go through a bunch of dramatic evolutionary changes more in depth than what we just did, from present day to the origin of life. And we're going to start with the Cenozoic, which is the age of mammals. That's from about 66 million years ago to present day. Then we're going to cover a little bit of the Mesozoic, which is the time period from about 250 million years ago to the extinction of the dinosaurs about 66 million years ago. And lastly, we're going to talk about the Paleozoic, which is everything else, all the other critters that aren't belonging to the Mesozoic or to the Cenozoic. And this is going to be the sort of biggest chunk of time, but also the evolution of many major forms is proposed to have happened during this time period. And we're going to consider lines of evidence from genetics, from ontogeny, from taxonomy, and from paleontology, as well as others as we go along. Now, I talked a lot about deep time last time, but I think that a lot of people don't really appreciate when we consider the proposed age for the universe, which people would propose the universe, like astronomers would propose the universe is about 14 billion years old. That's 13.8 billion years of time. That's insane. So to contextualize that a little bit, sometimes we use what's called the cosmic calendar. So basically it says, okay, if you were to condense the entire 14-ish billion years of history for the entire universe down into 1 year, when would stuff happen? And according to the cosmic calendar, we see the very first multicellular life on Earth in December. It isn't until December that we see the first multicellular life on Earth. The very first bones and shells show up December 17th. The very first amphibians show up the 22nd of December. The first mammals, the 26th of December. Flowers don't show up until the 28th of December. The extinction of the dinosaurs is around the 30th of December. And the evolution of virtually everything closely related to us happens on the very last day, with all of recorded human history being essentially during the last minute of the last day of December during the cosmic calendar, which is just like, it blows my mind. The last second would contain the scientific revolution all the way to present day, which is just a mind-bogglingly large amount of time that I cannot even begin to wrap my head around. But I think it's good to start with that because it sort of cements how much time actually exists to cover. We're going to start with the Cenozoic, right? So the major lineages that show up during the Cenozoic, or the age of mammals. There are a lot of weird animals that exist during the Cenozoic, and I'm going to talk about some of them, but here's some of my favorites. So on the bottom left, we have calicotheres. These guys are closely related to horses because they have an odd number of toes. They're basically gorilla horses. We have Thylacoleo, the marsupial lion, who has like these just bizarre scissor-like mouth, as well as opposable thumbs, and yet it's a marsupial. We have the entelodonts, the hell pigs, which were these enormous pig relatives that were like larger than modern day grizzly bears. Some of them were just absolutely enormous. Would have been terrifying to meet in person. There's Platybelodon down here, a relative of elephants, who their bottom jaws, their mandible, is basically just a shovel. And they use it to dig at roots, which you can tell by the wear patterns on those bottom teeth. And then, of course, because I study hominin evolution, human evolution, we have the likes of Australopithecus sediba, who would, as we'll sort of talk about later on, represents this really weird middle ground between what most people would consider non-human apes and modern day people. Virtually all of the major mammal orders of today evolved during the Cenozoic over the course of 65 million years. The earliest mammals that we know of existed alongside the dinosaurs during the Mesozoic. Some of them show up like early Jurassic, late Triassic, but they were very, very different from the types of mammals that you have today. First of all, they didn't, for the entirety of the reign of the dinosaurs, no mammal that we know of seems to have gotten much bigger than the badger size, right? Like these guys stayed small. And it makes sense because you wouldn't really want to be huge when you've got the whole world being run by dinosaurs, animals that are way, way, way bigger than you, and in many cases a lot faster and stronger. But after the extinction of the non-avian dinosaurs, tons of niches or ecological jobs and roles opened up for mammals to begin to diversify into. And so we're going to focus on the evolution of some of the strangest mammals over the course of the Cenozoic, or about 66-ish million years worth of time. During the Cenozoic, the world temperature fluctuated, but generally it was warmer early on, so like right after the extinction of the dinosaurs, and it's been mostly cooling off since then, leading to present day. And this idea is supported by like the geochemical work and paleo-environmental assemblages alike. I'll also note that mostly the continental organization looked like it does now. There are some differences though. Like if you look at this little map, you'll notice India is disconnected from everything else. And the way that we know this is because if you wind back, as we did last time for talking about the age of the Earth, if you wind back the movement of the continental plates, you can get their position at various periods of time. This too sets up a really nice prediction for where we should find specific animals based off of what was above and what was below water at any given moment. I've also got a picture of grass here because the Cenozoic is mostly when grasslands emerged. Like in the Paleozoic, grass hadn't even evolved yet. And during the Mesozoic, there's a little bit of grass, but really not that much. It's weird to think of a world without grass, but most of Earth's history has been grassless. And we know this because grass leaves behind these characteristic microfossils called phytoliths, and we don't find those, for example, during the Permian. Now, early mammals look pretty generic, while later, more modern mammals appear more specialized. This is a huge generalization, so don't at me mammal heads out there. But generally, if you look at the Paleocene, which is this epoch right after the dinosaurs go extinct, most of them just look like kind of radish-looking things of various sizes, whereas modern-day mammals are just displaying this incredible diversity in shape and form. So we have to explain a lot of different evolutionary emergences here. And Arctocyon is what that guy was based off of. That's the fossil that was drawn there. But I think it's fair to say that not all transitions are equal. So over here on the right, this is a phylogenetic tree of all of the major orders of mammal. So you have the monotremes, you have the proboscideans, like the elephants, you have saurinians, that's going to be like your manatees and your dugongs. We have scandentions, your tree shrews, primates, you have rodents, you have carnivores or carnivorans, bats, all this kind of stuff. And I think it's fair to say that if the first mammals that we have were pretty rat-like, for lack of a better term, it's going to be a lot easier to get modern rats than it is to get modern elephants. There's a lot more change, ostensibly, that has to happen to get a proboscidean from a proto-mammal than a rat from a proto-mammal, which is why I think I'd like to start off this tour through time with the most dramatic, in my opinion, of all mammalian transitions, which is cetaceans. Most mammals share the same basic formula. So here's our little proto-mammal here. I believe this is a drawing of Morganeucodon specifically, which is like an early-ish mammal for which we have some fossil material for. Most mammals have 4 legs, and those 4 legs have several digits. Most mammals have a tail of varying lengths. Sometimes it's shorter, sometimes it's longer, but usually it's a couple of vertebrae long. Most mammals have 4 types of teeth, right? These are characteristics that unite rodents and proboscideans and cats and antelope and chimpanzees and humans. Whales are different. Whales just like buck that trend completely, which is why we're going to start with them, or cetaceans. Modern cetaceans differ from other mammals skeletally in a myriad of different ways. So what you're looking at over here on the right is, I think that's a humpback whale or a mysticete whale. Then you have a dolphin, and then you have a tree shrew. So what you can hopefully see just by eyeballing them are cetaceans are super, super different from the tree shrew. And just for those of you out there who might not know all that much about cetacean evolution, all cetaceans today fall into one of 2 major groups: the odontocetes, or the toothed whales, and the mysticetes, or the baleen filter-feeding whales. And so you've got your baleen mysticete up here, and then below you have your odontocete. And like, these guys have these fully aquatic fusiform bodies. Fusiform means it's sort of shaped like a missile. It's like really hydrodynamic. Incidentally, this is why we design submarines the way that we do. They don't have any external hind limbs. Their elbow is immobile. So like they have this tiny little elbow up here, right? There's the humerus, or the bone of the upper arm, and then there's the radius and the ulna, but they can't like flex it like how a human can, nor can they supinate and pronate their wrist at ease. Like they can't cross those forearm bones. They have dorsal nares or nostrils. That's their blowhole. They also have a feature called an involucrum, or an involucrum, which is sometimes what people call it. This is a feature of their hearing system, of their auditory system, a feature specifically of the tympanic bulla. And it is, quote, from Marks, Lambert, and Uhen 2016, the clearest trait uniting all living cetaceans is a marked increase in the thickness and density, and that's called the pachyosteosclerosis, of the medial wall or middle side wall of the tympanic bulla. So it's basically, as you'll see in a second when I show you a picture, this big thickened chunk of bone that helps pull the inner ear away from the skull and that aids them in hearing underwater. And what's critical about the involucrum is no other living animal group has it, even other ones that live underwater. Manatees and dugongs don't have it. Seals don't have it. Otters don't have it. It is totally unique to cetaceans. They also have a fluked tail, which means their tail sort of bars off into 2. And they have homodont teeth, or baleen, which means that all of their teeth look the same. So there's that fusiform or streamlined body shape. This is what the involucrum actually looks like. So here's where it sits inside a dolphin, sort of back by their mandible, like right where their ear hole is. And here's me holding one and pointing to the thickened wall there. That's a whale involucrum that I'm holding. It's basically, again, this big thickened side of the portion of the tympanic bulla for hearing underwater. There's the homodont teeth. So you can see in the mouth of this dolphin here, all of the teeth look the same. This is not how you and I are. If you take your tongue and probe it around in your mouth, you can find that you've got molars in the back, and then you've got your spiky premolars for shearing, and then you've got your pokey canines for stabbing, and then you've got your chisely incisors for biting and for breaking pieces off of whatever it is you're eating. And incidentally, like most mammals have those 4 types of teeth, but not cetaceans. And here's the flukes. So what's cool about the flukes is we can actually estimate whether or not fossils had flukes or not, even though the flukes don't preserve, because in order to actually hold the weight of a fluke, the vertebrae in the caudal section of the vertebral column taper in a very specific way. And the way that we sort of clock how this works is that manatees and dugongs, who are also aquatic mammals, manatees have this paddle-like tail and dugongs have a fluked tail. So we can actually like chart the width and height of each of their individual vertebral bodies and plot that in something without a fluke versus something with a fluke, and then compare that to a dolphin and get the pattern for what fluked tails look like, even if the fluke isn't there. Does it make sense how that works?
Mm-hmm. Yep.
Cool.
Hey, did you know that there are whales we've heard and seen carcasses of, but have never seen alive?
I did not know that, but that doesn't surprise me because the ocean is huge and very scary.
Yeah,
Like
it's so cool to me. There's one that we saw alive for the first time about a year ago.
a year?
Yeah, it's the ginkgo-toothed beaked whale was seen alive for the first time about a year ago.
Hold on, I'm writing that down because I want to see what it looks like. Ginkgo-toothed
Beaked
beaked
whale.
whale. I actually looked up pictures of beaked whales for this presentation, and they're freaky. I had no idea that there were whales out there that have like tusks like that outside of narwhal, which they have just the one, and it's usually only in males. But beaked whales, they've got like these two tusks that curl up on either side, and they use them, at least we think, the males are using them to compete for access to females because that's usually why tusks show up. But like they live in pretty deep water, and I'm just imagining like you're a poor lone beaked whale, and you like a challenger approaches, in the depths, and you've got to like duke it out for access to the first female you've seen in like 6 months. I feel for these guys. But that doesn't surprise me. I've actually heard from people who study like invertebrates, like specifically squid, like the squid people will tell you all about how the size of some of the squids that we're seeing are probably underestimates because the ones that get really, really big don't tend to die and then come to the surface. So I don't know. I don't know how that's going to impact our estimates. I would bet that giant squid and colossal squid probably get a little bit bigger than we're currently giving them credit for. Probably most species of abyssal squid, but it's freaky.
It is. I love the ocean.
Me too, but I love it in a way where I mostly fear it. Okay, outside of the skeleton, cetaceans are even weirder, right? They have almost no hair. They have baleen instead of teeth, at least in the case of mysticetes. And what's weird about baleen is that it's not a tooth. That was another thing that I learned over the course of researching for this. Baleen is actually more similar to hair than it is to teeth. They also echolocate and or sing. So mostly you see echolocation in the toothed whales, and you mostly see intense, like those beautiful songs in the mysticetes, but not always. And all cetaceans have unique adaptations to diving, with the ones that dive really deep having even more specializations physiologically. They can also get huge. The modern day blue whale, female blue whales today are the biggest animals that we know of to have ever existed. That blew my mind. That female blue whales, we know of no animal that gets bigger than that, even in the fossil record. Crazy. So that's all very weird. If cetaceans evolved from land mammals, that should be really obvious. We should be able to show this in stunning detail. So first, we got to start with the genetics. This is crazy. This is going to blow a lot of people's mind. But genetically, all cetaceans nest with a group called the artiodactyls, or the even-toed, hoofed mammals. And their closest living relatives are the hippopotamuses. So here are some artiodactyls: camels, giraffes, deer, zebra, ibex, camels, bison. All of these guys have an even number of toes, whereas perissodactyls, which is the odd-toed ungulates, have an odd number of toes, and that group would include things like horses and rhinoceros and tapir. But cetaceans are most similar genetically to the artiodactyls, or the even-toed mammals, and the one that they are most similar to, genetically speaking, are the hippopotamuses. So here are some papers that will sort of display that for you. You've got molecular evidence for the inclusion of cetaceans within the order Artiodactyla, and then this much longer title underneath it, specifically singling out hippopotamuses as the closest living relatives. Okay, if that's true, if cetaceans' closest living relatives are artiodactyls, even-toed hoofed mammals, this should be obvious and evident from the fossil record and from development of cetaceans. So,
real quick.
yes, please.
If I recall, the hippogenetic link is fairly recent and was kind of a surprise. And I think hippos were not necessarily nested where they are now prior to understanding their genome.
I don't know. I know this first paper is from the 1990s, but I don't know about the second one. If I had to guess, certainly last 20 years. Like certainly last 20 years, probably if I had to guess, maybe last 15, maybe even last 10. So I agree. And I think it was surprising because hippos are pretty derived for artiodactyls, but yes, let's go with yes. I believe you.
Okay, cool.
All right. So the cetacean fossil record. Cetaceans are proposed, according to the fossil record, to have evolved over around 10 million years of time during the Eocene, when generally speaking, the world was warmer and the sea levels were higher than they are today. The fossil cetaceans leading to modern cetaceans are known as the archaeocetes, which makes sense because archaeo means old or primitive or ancient, and cete means whale. So these are the ancient whales. All archaeocetes are transitional in the sense that they occupy this temporal and morphologic midway point between modern whales and some kind of ambiguous hoofed ancestor that we'll talk about later that lived on land. But as far as I know, none of the fossils that we're going to be talking about today are like proposed to be the common ancestor of modern cetaceans. Instead, as you can see in this sort of chart over here, it's something that's representing like a potential ancestor. And the reason is because we can never know for sure. Like it could be the ancestor or it could be something really similar to the ancestor. But without the genetics, we can't say this for sure was the one. We can only say that this is a part of the same group as the one that was the last common ancestor. These fossil whales that we're about to talk to demonstrably show, I'm going to argue, the emergence of fully modern marine cetaceans from extinct terrestrial relatives. So the first group of whales that we're going to talk about are the neocetes. Right, so here are our living whales and dolphins. We have our mysticetes on the left, a picture of one, and then its skeleton. And then we have a dolphin and a dolphin skeleton there on the right. Here are two neocetes from 11 million years ago and from 18 million years ago in Cetatherium and Wimmal, respectively. Now, just eyeballing it, yeah, those look like whales to me. That tracks. Here's when the neocetes lived from about 34 million years ago to present day. And the reason is because since the whales first emerged during the Eocene, after the Eocene, the whales are here and they are diversifying. So the neocetes whales that we're going to be talking about are like ancient, mostly extinct whales, but they are technically related or a part of the neocete group, which modern whales also belong to. This is kind of like how like a saber-toothed cat would be a felid, even though we don't have any modern saber-toothed cats. So where in the world did the neocetes live? Here is a sort of graphic depiction of what the world looked like during the late Eocene, about 36 million years ago. Mostly the same as it does today, although you'll notice in this sort of middle Eocene picture here on the right that like India hasn't fully connected just yet. This over here on the right is like a modern, like where they're found right now. And then here's what the world actually looked like on the right. And you'll see that there's this big seaway that's cresting into South America and like Florida's underwater, but still like familiar. And neocetes are found all over the world because modern whales are found all over the world. Modern cetaceans, a lot of them have these very large ranges because they can swim all over, they can go wherever they want. The neocetes include living groups, Odontoceti and Mysticeti, along with some extinct members of these groups. Here are a couple of fossil crania of each of these groups. And I'll tell you, Will, like from here and also moving forward, anytime that I show you a species, a proposed species, or a proposed transitional species, my promise to you is I'm never going to show you a drawing unless you can also see the actual bone material. And the reason is because drawings are great. They make things clearer, but I think it's really important that people see the original material as well so we know what we actually have.
Thank you.
Of course. Neocetes ranged in size from 4 feet to 79 feet long. They were fully aquatic. They had no external hind limbs. Their elbow is immobile and they couldn't supinate it, just like modern whales can't. They had dorsal nares or nostrils on the tops of their heads. They had an involucrum. They had a fluked tail, and they have mostly homodont teeth or baleen. So, like, these are cetaceans. That much is clear. Here's a bunch of cool crania to show you some of the mysticetes on the left and the odontocetes there on the right. And part of the reason that we can clock them as cetaceans outside of just observing them is, again, they have that involucrum that only whales have, at least so far as we can tell. However, these guys are really interesting because it is in neocetes that we see the evolution of baleen and the evolution of echolocation. And we can watch these abilities or characteristics show up through the fossil record. So in Yanocetus here, we see Yanocetus, Adiocetus, and Yamatocetus there over on the left. We can see baleen emerges first alongside teeth before eventually taking on the feeding emphasis and replacing the teeth entirely. So in Yanocetus, there's only teeth at 38 million years ago. Sure, makes sense. 4 million years later in Adiocetus, these guys had teeth and baleen. And we know that they had both because we can look at their jaws, and baleen and teeth leave specific scars or sockets on the mandibles and on the maxilla that are characteristic. And we know because we have toothed whales and whales with baleen today, so we can compare them. So Adiocetus had teeth and baleen, but they were mostly using their teeth. Their baleen was small and maybe a little bit helpful, but it was nothing like the kind of baleen that we see in a humpback today. Yamatocetus had teeth and baleen too, but now the positions of the teeth and the baleen, as far as emphasis goes, have switched. And in Yamatocetus, you can see in this mandible here, there's no teeth or baleen because baleen doesn't fossilize very well. But like the teeth are basically just like you can tell on the basis of the size of the roots that are inside these mandibles. They're small and rudimentary. They're not using them for much, evidently. So we see no baleen to some baleen to mostly baleen, and then in modern whales, only baleen. Modern mysticetes, I should say. What's cool about this is that mysticetes today still have all the genes to make teeth. They have them. They're still sitting in their genome, and we can clock them because we know that these genes, when they're messed up, result in abnormally or non-developed teeth in toothed whales. The mysticetes have these genes too, but in mysticetes, they're turned off. They are pseudogenes. They can't access them. What's cool about this is that development of these teeth actually starts when the whales are in utero, but never completes. The teeth start to form, and typically they're reabsorbed before the baby is born, but not always. And sometimes when babies, like baby mysticetes are born and they don't survive, like they wash up on the shore, or this happens, unfortunately, in captivity, we can actually dissect away the portion of the maxilla that is closed off, and you can see these teeth like mostly or fully developed in the crypt. They'll never erupt, and they don't have any enamel, but they're teeth sitting there, a remnant. Likewise, echolocation can be seen showing up in our odontocete whales, because dolphins are all whales. That was another thing that I learned. Not all whales are dolphins, but all dolphins are whales. And the way that we know this is because echolocation, in order for it to be carried out like how it is today, it requires a specialized organ called a melon, which sits at the sort of rostral portion of the face on these little guys, like in this bottlenose dolphin here. So there's the brain, there's the melon. And the purpose of the melon is it's like this big fatty organ that helps direct sound so that they can hear their little clicks that they make. What this means is that the melon organ can act as a proxy for modern echolocation. And as we move through time from Squalodon to Y. piata to Leviathan, we can see the concavity of that area where the melon should be emerge over time. All of that said, ancient neocetes are just cetaceans, but some of them are kind of primitive, right? Like Squalodon, this is like a dolphin-y looking thing, even if it doesn't have the full melon organ or really any melon organ at all. And Adiocetus and Yanocetus, these guys are mysticetes, even if they don't have the full-on baleen, but like they're whales. Like this is not controversial. Anybody could look at these and say that these are whales. Check. Moving back a little bit deeper in time, we have the basilosaurids, or the king lizards. That's what their name means. And the reason is because when these fossils were first discovered, people thought they belonged to dinosaurs. Basilosaurids lived from about 34 to 43 million years ago. They too are found all over the place because they were fully aquatic. And basilosaurids also got really big. They were 8 to 66 feet long. They were also fully aquatic, but they had tiny external hind limbs. So hind limbs, but they weren't doing much. The elbow, is mobile, so they actually could move that elbow a little bit, but they still couldn't supinate it. They also had intermediate nares or nostrils, not a blowhole on the top of the head. They have an involucrum. They had a fluked tail, and they have mostly homodont teeth, but not completely. So you can see in Dorodon over here to the right, you've got this sort of serrated edge there on the back teeth, and the foreteeth lack that serrated edge. So they're not all quite identical to each other. Taking a step back, I want to talk a little bit about the acoustic system, and again, that involucrum for these cetaceans. Modern day cetaceans don't just have that involucrum, they actually have several different adaptations of the inner ear that allow them to hear underwater. And one of those characteristics is a fat pad that sits in this depression on the medial side of their mandibles. So if you feel on the inside side of their lower jaw, you'll see this depression in modern dolphins, and a big fatty pad sits there that helps conduct sound. Here is that little indentation, that little fatty pad depression in Dorodon. So we know that basilosaurids also had at least a little bit of that fatty pad. And then here's a better, more scientific look at where that fatty pad is in a cortical bone look. Hearing is crazy. Hearing is insane to me. I was reading a lot more about hearing because I was trying to understand the importance of this system in this animal group. And I've included this GIF here to show how hearing works. Basically, sound is waves. Sound comes into your ear as waves and sort of vibrates the tympanic membrane or your eardrum. But your eardrum articulates with 3 auditory ossicles or inner ear bones. And these are the malleus, the incus, and the stapes. Those guys articulate with each other to bang essentially on the oval window, which is right over here indicated by my cursor. It's this thin membrane. And that membrane sends the vibrations down this snail-like organ called the cochlea. And the cochlea is filled with fluid as well as tiny hair cells. And these hair cells pick up on the vibrations in the fluid and convert those mechanical signals into electrical signals. It is insane how this actually works. So this is what I just said. The 3 auditory ossicles in mammals articulate with one another to send the signals from the eardrum to the cochlea, where the hair cells convert the waves into electrical signals. But cetaceans are specialized for hearing underwater. So their auditory ossicles are going to be different than ours are, right? Like hearing underwater and hearing on land, these are 2 different things. This is why every single one of us who's played the game where you get with your friends underwater when you're a kid and you guys are like trying to tell what the other person is saying, this is why you can't hear it very well, because water conducts sound differently. And the way that sound is conducted in your head when you're underwater is slightly different than when you are above water. Here is a cool little comparison of modern day baleen whales or mysticetes with our toothed whales or odontocetes. And you can see the malleus is this big bloated monstrosity. The incus is this kind of L-shaped bone that's sitting on like a little stand. And the stapes or stirrup is mostly the same. And it's similar between these 2 groups, although the stapes seems to differ a little bit. Basilosaurids, it looks the same as what you would expect for a whale. So their little inner ear bones are whale-esque. So are basilosaurids cetaceans? Yes, they are. However, basilosaurids, while they're more similar to neocetes than they are to anything else, they're fully aquatic. They've got those large fusiform bodies. They've got the involucrum, the fluke tails. They could move their elbows a little bit. Here's a comparison of a modern day whale versus Dorodon. And what you can see is that where the humerus or that upper arm bone articulates with the radius and ulna, there's a lot more space in Dorodon, this lower picture, than there is in modern whales. So they could actually flex their elbow just a little bit. Likewise, basilosaurids had slight heterodonty, so they had some different types of teeth. can see again there, that's the actual cranium. Basilosaurids also lack a blowhole, so their nose is more forwardly placed compared to what we see in the Neocetes. But most strikingly, Basilosaurids have hind limbs. Now, these things were very small, but they were external from the body. That means they poked out actually outside of the body wall. They had a bony pelvis and hind limbs with all the same bones that modern day mammals have. So here's a rat hind limb, and you can see there's the pelvis, there's the femur, there's the tibia and the fibula, and then in the rat, that foot is flexed forward, whereas in this Basilosaurus, it's flexed backwards. But as you can see in this big Basilosaurus skeleton that's hanging from the ceiling, this pelvis did not articulate directly with the vertebral column. Instead, it connected via cartilage. So it wasn't, these guys could not walk on land. Modern day whales do, in some species, actually have a remnant of what we see in these Basilosaurids. They have a bony pelvis. And like in Basilosaurids, it is not at all connected to the rest of their body. It's cartilaginous at best. And the purpose of the pelvis maintaining in these larger mysticetes is that it actually helps anchor the penis. It helps them mate. So there's a purpose for this structure being maintained in some modern cetaceans. But obviously, as you can see in these bones, as articulated over here with the pelvis and the femur, this is the whole thing. This is all they've got. This is way less complex than what we see in Basilosaurus, who has the full pelvis, the full femur, the full tibia, the full fibula, all of the ankle bones and toes. Like they have a full leg, even though it's not doing much. So we're seeing the loss of legs from Basilosaurus to Neocetes over the course of a couple of million years, but there's something much more important lurking in this little unassuming hind leg. Modern-day artiodactyls, or our even-toed hoofed mammals, have a ton of traits that identify them compared to other mammalian orders. So remember I told you earlier that carnivorans have those carnassial teeth, and no other mammal group has those. So if you have those, that's what tells us that you're actually a carnivoran. The same is true with artiodactyls. And one of the characteristics that defines artiodactyls is a double pulley structure in their ankles called an astragalus. And what this does is it provides them stability so that they can run and turn at these really high speeds. So you've probably heard that horses break their legs really easily. Artiodactyls don't have this problem. Their leg system, their running system is just so tight and stable that they can move on a dime and they don't have to worry about pulling or breaking anything or popping anything out of any sockets. And part of the reason for that, at least in the ankles, is thanks to this astragalus, which you can hopefully see right there. Today, only artiodactyls have this double pulley system. That's it. Like this is a defining feature of artiodactyls. Horses don't have it. Pigs are artiodactyls. So like horses don't have it. Carnivorans don't have it. Like humans don't have it. Just artiodactyls. But Basilosaurus has this astragalus. It has an artiodactyl ankle bone, despite the fact that it is fully marine and its legs don't work anymore. Remember, the closest relatives genetically, according to genetics, to cetaceans are artiodactyls. And only artiodactyls have this. And then here it is. We found it in Basilosaurus. Hippos have this too, for the record. Why do they have it? And I think this leads us to a better question. Why did cetaceans lose their hind legs at all? So modern cetaceans actually begin development with their hind limbs. So here are a bunch of really cool papers that note it, but I like this one because it actually shows you where those hind limb buds are. So A is a pantropical spotted dolphin, and B is that same embryo later on in development. And you can see early, here they have a hind limb bud that's beginning to develop, right? And then it is reabsorbed back into the body. But sometimes it doesn't reabsorb. In super rare cases like this Japanese dolphin named Haruka, cetaceans can be born with their hind flippers, and their hind flippers have bones in them. And this is the only image. I was so interested in this case because it was mentioned in my Walking Whales book, and then it was also mentioned in it's basically mentioned in a lot of developmental case studies on cetaceans. But this dolphin is Japanese, and the people who own her, and then she died in 2013. So the people who own access to her body are notoriously weird about letting people come and like CT scan it. So this is the only X-ray that I was able to find, but you can see the individual digits on this hind flipper. At least that's what it looks like to me. We understand how this happens. So this is an abstract from this paper that I have over here on the side, Developmental Basis for Hind Limb Loss in Dolphins and the Origin of the Cetacean Body Plan. And I'm going to read the highlighted section for you here. Our data indicate that cetacean hind limb buds form an apical ectodermal region, so a bud, and that this structure expresses FGF8, so the gene that helps them actually develop the limbs initially, but that neither the AER or FGF8 expression is maintained. So the limb bud starts to develop and then it terminates. Like we already knew that, cool. They say, moreover, sonic hedgehog, which is a specific Hox gene that you were reading about earlier, watching videos with Sean Carroll earlier in this series, which mediates the signaling activity of the ZPA, that's the zone of polarizing activity, is absent from the dolphin hind limb bud. So basically the signals start, but because the director of those signals is absent, they terminate and they can't actually complete. We find that a failure to establish a zone of polarizing activity is associated with the absence of HAN2, another of our body plan genes, an upstream regulator of sonic hedgehog. Interpreting our results in the context of both the cetacean fossil record and the known functions of sonic hedgehog suggests that the reduction of sonic hedgehog expression may have occurred about 41 million years ago and led to the loss of distal limb elements. The total loss of sonic hedgehog expression may account for the further loss of hind limb elements that occurred near the origin of modern suborders of cetaceans about 34 million years ago. Integration of paleontological and developmental data suggests that hind limb size was reduced by gradually operating microevolutionary changes. That's so cool. Like they did this by basically going to these museums that have all of these embryos preserved like for hundreds of years, or not hundreds plural, but for like 100 years in some cases, stored in formaldehyde. And they're like taking these microscopic slices and then looking at what's actually active in each of those slices. And these, as you can see in these microscopic pictures over here. I'll also add that both unique and convergent adaptations, speaking of genes, that allowed basilosaurids and neocetes to dive as deep as they do, these emerged in some of the cetaceans, but not all of them, which is really interesting. And we've managed to find that in some cases it's the same gene and in some it's different. So in these two papers here, Into the Blue and then Evolutionary Genetics, they note that we have 42 genes associated with pulmonary fibrosis across 45 mammalian species. So these are the ones that they investigated. And 21 of those genes in cetaceans exhibited accelerated evolution, featuring specific amino acid substitutions and 14 of them. So what that sentence is telling you is that of the 42 genes associated with basically like breathing and cardiovascular health in mammals that live on land, 21 of them, those versions in cetaceans are different. They've evolved specific mutations that give them unique characteristics. So these genes exist in terrestrial mammals, but they're different in cetaceans, and that's what gives them the ability to hold their breath longer and not get decompression sickness, etc. They say primarily linked to the development of the respiratory system and lung morphological construction, these genes play a crucial role. Moreover, among marine mammals, we identified 8 genes undergoing positive selection, and the evolutionary rates of 3 genes significantly correlated with diving depth. And so they're basically like saying which genes are actually responsible, and they were able to correspond 3 separate genes with diving depth, not just the ability to dive deep, but the depth specifically that they are going. I don't know how they're doing this kind of stuff. It's really cool, but it's definitely above my pay grade. So in sum, basilosaurids are cetaceans, but they're distinctly primitive compared to the neocetes. They've got all the cetacean characteristics, but they also have some weird stuff about them. Next up is Protocetidae, the before whales. Now, these are the first guys that you look at and you're like, that doesn't look like a whale to me. Here's when they live, older than the basilosaurids, but overlapping with them from 35 to 47 million years ago. Where are they living? Also worldwide, but only some of them. And that's going to be interesting because the older ones that are better adapted to the aquatic habitats are the ones that are found worldwide. And the early ones are not. They're limited to this region here, Indo-Pakistan. So here's protocetids. We have a lot of them. They're pretty cool. They're small. So the reason I talked about some characteristic, or why I'm stopping and talking about genetics as we go, is to say, okay, which genes are responsible for these characteristics that define modern cetaceans. These guys are little. They don't grow to the massive sizes of modern mysticetes. They're not getting much bigger than 15 feet. They are mostly aquatic, but they have large hind limbs, not like Basilosaurus. Their hind limbs are actually pretty big, and they vary in how functional they are. Their elbow is mobile, but they still can't supinate it. They have forward and intermediate nostrils depending on the protocoetid in question. There's the more forward nostril, and there's the intermediate nostril. So Artiocetus, the younger of these two individuals, looks more like the Basilosaurids, and the older individual has that regular nose sort of on the tip of the face that you see for most mammals. Protocoetids have an involucrum. They also had paddled or fluked tails, but it depends on the specimen and it depends on who you talk to. Heterodont teeth. So they've got specialized teeth for specialized jobs like most mammals and not like modern cetaceans. Just like Basilosaurids, these protocoetids have that double pulley ankle that you see in artiodactyls. So over here to the right, this is an astragalus found on the savanna. I'm not sure which type of artiodactyl it actually belongs to, but it's got this characteristic double pulley on both sides. Over here to the left, you can see Rhodocetus, which is a type of protocoetid whale, and Artiocetus, which is another type of protocoetid whale, compared to a pronghorn antelope. And like, whoa, I just smacked my mic. I got too excited. I just love cetaceans. Spitting image. You know what I mean? These things are very, very similar. Same type of bone. And then just for comparison, down here, you can see the difference of other artiodactyls like pigs and deer. And all 5 of these guys that I'm circling with my cursor right now are different from what you see in a dog. Here's another astragalus from an artiodactyl called, a cetacean called Perugocetus, who has that same double pulley. Like this is a great evolutionary expectation. We should, if evolution is BS, we should be able to find one whale that doesn't have a deer's ankle. Here's what's weird about these guys, though. Some protocoetids also had tiny hooves, and I learned this over the course of researching this. So you can actually clock whether or not an animal has hooves or claws based off of, or nails for that matter, based off of what the tips of their phalanges look like. So your phalanges are your fingers and your toes. And in the case of claws, the tip of the most distal or like the furthest out phalange will have space for a nail, or it'll actually have that nail or claw. And the bed of that bone will have a specific type of depression for it. In the case of hooves, as you can see in this pig here, the hoof, or the singular hoof, actually wraps around the entirety of that distal phalange, that distal phalanx. And this makes sense, right? Like when a dog walks around, they're not walking on their claws stick off the edge of their toe. When we walk around, our nails aren't touching the ground, hopefully, they're on the top of our toe. But horses and cows, they're walking on that hoof. That hoof wraps all the way around the distal end of the phalange. And we can actually tell, looking at the structure of the bone, whether or not a hoof was present. So the way that we tell, as you can hopefully see from there, is there's sort of like a longitudinal groove and then sort of a concave shape. There will be a better picture here in a second to maybe help. Protocoetids, this is Myocetus, this individual here at the beginning, right here, Myocetus, that individual there, has little phalanges that had hooves on them. You can see the longitudinal groove. You can see that sort of spade shape there at the tip of the toe, as did Perugocetus. So both of these protocoetid whales had fingers that ended in hooves, even though they barely ever came to shore, although these guys did come to shore. Here's a better comparison. There's Myocetus, a hoofed cetacean. There's the digits. I've flopped the picture over so you can see a little bit better. Here is Mesohippus, a hoofed horse relative. You can see I picked Mesohippus because that middle toe is so big. And you can see that middle hoofed toe spitting image of the individual toes of Myocetus. Same with Cynonyx, which is a hoofed mesonychid, whose toes similarly have that sort of spadish shape or that hourglass shape. And they have that longitudinal groove there in the middle. So protocoetids were artiodactyl-like in having that astragalus and in having little hoofed toes. But like cetaceans, they had an involucrum. So not just an involucrum. They didn't just have that thickening of that inner ear bone that, again, only modern cetaceans have. But as you can see in these two figures over here to the right, they're semicircular canals, which are these little canals that exist in all mammals that help us keep our balance. The semicircular canals look more like cetaceans than they do anything else. So in the red box is a protocetid. In D, which is I'm circling here with my cursor, this is a modern bottlenose dolphin. In B is another earlier whale that we'll talk about next. And in A, this is just a galago, like a primate. And here we have Myocetus. It looks most similar to cetaceans. And again, it has that involucrum, and it has all of these other, you can stop and look at figure 5.8, or excuse me, 5.10 here later if you want, and see all of the other tiny characteristics of the inner ear that tell us Myocetus has a whale's ear. But they have more transitional features than just that. We have this insane fossil of Myocetus. It's a female, and it still has its baby in its belly, right? So it's the skeleton, and it's imprinted in the stone, and then right in the abdominal region, there's a fetus of the baby Myocetus. And what's cool is that the way that this baby is oriented is the head is up, and the feet are towards the vaginal opening, where the vaginal opening would be for Myocetus, which means these guys gave birth, or excuse me, opposite. The head is facing towards the vaginal opening, and the feet are facing away, because these guys gave birth head first, like all land mammals do, like humans do in most situations. And the reason why whales don't give birth like this, because whales give birth tail first, is because they want to give them the maximum amount of time to actually fully get through the birth canal before they're forced to race to the surface to take their first breath, before the umbilical cord detaches. And so these guys gave birth like land mammals, not like cetaceans. They also had limbs articulating with the pelvic complex and with the spine that could bear weight, and at least some of them did. And they had large feet, so when they came to land, they would have been kind of clumsy. Underwater hearing is further facilitated by that fat ped that I talked about earlier. And they also have reduced turbinates. So, dogs have like wet noses. Dogs also have, as you can see in the bottom left here, nasal turbinates. And in fact, most mammals have this. These are these scrolls that are like rolled up in the nasal cavity, and they increase the surface area for smell particles to bind and then for you to pick up a signal. Humans, we have them, but we don't have a ton of them. Primates in general don't rely very much on smelling, but dogs, cows, hippos, horses, most mammals have really in-depth turbinates. Myocetus did not have nasal turbinates that were very impressive. It looks more like modern whales in this way. And this makes sense because you can't smell underwater, at least not with an air setup. Smelling is super important to land mammals, but in modern cetaceans, most or all the genes are there, but off. This one blew my mind. I thought this was crazy. In this paper here, Aquatic Adaptation, the Evolution of Smell and Taste in Whales, you can see just like almost like nearly 1,000 genes devoted to smelling and chemoreception in general in this cow versus like less than 100 in every kind of whale, and as few as like 13 in dolphins, like that are on. Like the genes are there, but they're off. It's crazy. And Myocetus, like modern cetaceans, seem to have had very little in the way of the mechanics for smelling. We don't have their genes, but it doesn't seem like it was very important because they don't have those turbinates that cows and dogs, etc. have. This also makes the olfactory genes that they have, just like the genes that we talked about earlier for growing limbs, pseudogenes. This one was crazy to me. So Myocetus, who I just talked about, who comes onto land to give birth, they were capable of walking on land, but they would have been really clumsy, kind of like a seal, right? That's the skeleton there. So you can see those hind limbs. It's the bottom one. Myocetus is the lower one. It's got those hind limbs. It would have been able to come on land and give birth, maybe escape from predators, things like that. And the reason we know that they could walk on land outside of the fact that they have legs is that their pelvis actually articulates with that vertebral column. Right? In humans, we have a sacrum. It's a set of sacral vertebrae in the back, and that's what either half of our pelvis fuses to, and that gives us our pelvic girdle. And that's the case in every land mammal that moves around on all fours. It's also the case in Myocetus. But in modern cetaceans, if you will recall, in like bowhead whales, they have that pelvis, but it just floats cartilaginously in their body. Same with basilosaurids, same with most neocetes. Myocetus has that pelvis, but it's attached. But the later protocetid, Georgocetus, has the same pelvis but detached and no sacrum. So in 7 million years, the ability to walk on land, like walk competently on land, disappears in this group of animals, in protocetids. So Georgocetus couldn't bear any weight on its hind limbs, or if it could, it would have been like even clumsier than like a modern day seal, whereas Myocetus could have gotten around just fine. Here's a good question. Why did these guys stay small? Like Georgocetus and Myocetus, again, they're not getting to be like 15 feet long, whereas modern day blue whales and also basilosaurids could get really, really big, like 80 feet. Modern work implicates 4 specific genes as being responsible for why whales can get so huge. What's cool is that each of these genes, at least insofar as I could understand this paper, are present in close relatives of whales, but they're again altered in cetaceans. So again, just like we saw with smelling or with limb loss, they're in regular mammals, but they're different. They've mutated in cetaceans. I really like this abstract, and I won't read it for the sake of time, but if you pause it, they talk about how the genes that are linked to these huge sizes in the likes of blue whales are also involved in mitigating the consequences of large sizes. So they're also involved in like repressing cancer and in cardiovascular health and a lot of things that can be impacted if you get too huge, which that makes sense. Like the mutation wouldn't show up if you were getting big, but also getting cancer every 5 seconds, you'd be selected again. So it has to be something that impacts several areas at once, as we tend to see happen in evolution a lot. These supersized whales of today didn't come around until about 5 million years ago, like blue whale sized anyways, which is another thing that came from this paper. Okay, next group of whales. These guys don't really look like whales. These guys are called Remingtonicidids. There are not very many species of them, so I won't spend too much time on them. They lived a little bit earlier than Protocetidae, and as I hope you can appreciate here, just like with our dogs descend from wolves, but wolves are still around example, you see that these groups overlap, but that as we move backwards in time to more primitive whales, the oldest fossil always belongs to the most recent group, to the oldest group in question. Remingtonicidids are the first group of cetaceans that are really limited in where they're found. They're basically only in Indo-Pakistan and Egypt as well. And the habitats that they're found in is mostly swamps and marshes. These guys were similar in size to the smaller protocetids. They were mostly aquatic. They had large hind limbs that varied in functionality, although older ones could move on land more easily than younger ones could. The elbow is mobile, still no supination. They have forward nostrils. None of these guys have anything even close to a blowhole. They have an involucrum. They have a paddle-like tail, and they have heterodont teeth. They're kind of like, the way that I have seen these guys described is they're kind of like a more land-biased Protocetidae. These guys are thought to have occupied really similar niches to modern-day river dolphins. And the reason why we say this is because their anatomy is really similar to river dolphins, craniodentally speaking. They have these little itty-bitty eyes, and they have these big pits on the fronts of their noses where nerve bundles for enormous whiskers would have been. And we know this because modern-day animals with really big whiskers have these big nerve pits. And so these guys likely were living in these marshes and using those whiskers to sense changes in the water and to know sort of where to go, where the prey is, things like that, kind of like modern river dolphins do with echolocation. They're sort of like an otter version of a river dolphin. Like we saw with Protocetus, some of these guys could walk really comfortably on land, like Dalanestes, and others could not, like Remingtonicidius. What I think is really cool is that modern-day cetaceans do have a tiny bit of hair when they're born, and it's in the same spot that Remingtonicidius had these huge whiskers, right? Like it's right at the tip of that nose, and it's just barely a few teeny tiny little hairs, and they usually don't keep them. Speaking of which, hair loss in cetaceans is linked to their offshore, fully marine lifestyle because hair is just going to increase drag. Like it can stick around in seals because seals come to land, right? But since cetaceans are our only fully committed marine guys, they've lost hair, like all of their hair. And similarly, dugongs and manatees have lost most of their hair. But what's cool is that we've actually isolated the genes that are responsible not just for hair loss in cetaceans, because remember, just like with all the previous things, they have the genes for the hair, but they're busted, they're pseudogenes. But we also found the genes that are responsible for their thermoregulatory replacement, which is blubber. All mammals have subcutaneous fat, or fat that's just underneath the skin. Blubber is just a supercharged version of that. So evolving blubber didn't involve evolving any new structure, but an elaboration on an existing one. So protocetids are cetaceans. We count them as cetaceans primarily because of that involucrum, but they're still really, really primitive compared to basilosaurids. Ambulocetus is our next group, the walking whales. Ambulocetus is found, again, even older than anything previous. Where are they? Indo-Pakistan. That's where they're stuck. And that's going to be where everything else is from here on out. Ambulocetids are really cool and interesting primarily because we know a lot about them, because we've got this really marvelous, nearly complete skeleton. They were decently big, but nothing close to modern cetaceans, about 10 feet long. They were terrestrial and aquatic. They had powerful hind limbs and forelimbs. The elbow is mobile, but they still can't supinate it. So like they're still not able to do this. They have forward-facing nostrils. They have an involucrum. They have a paddle-like tail and heterodont teeth. So the main things that's linking these guys linking these guys to cetaceans is that inner ear, the involucrum and other structures of the inner ear, and the immobility of that elbow. Shocker, they have astragalus, just like everything else prior. And also shocker, just like everything else prior, their little toes are tipped in hooves. So these guys are artiodactyls, like for sure. Just like every subsequent cetacean, Ambulocetus has an artiodactyl double polyastragalus. Likewise, their toes are tipped in small hooves as seen in earlier protocetids like Myocetus, Rhodocetus, Artiocetus, and Perugocetus. And then there's just, if you want to pause and like check this out later, you can see for yourself. But just like cetaceans, they have that involucrum, and not just the involucrum, they have that fat pad. They have that mandibular foramen for where that fat pad sits in later cetaceans to help transmit that sound, because these are spending a ton of time actually in the water. There it is in the actual fossil. And these guys had semicircular canals that, just like we saw with some of these other protocetids and the like, was more similar to cetaceans than it is any land mammal, which is weird because these guys are spending a ton of time on land. So we count them as cetaceans because they have these cetacean characteristics. These are like the first guys out of anything we've ever found that are spending the most time on land, but they still have that whale inner ear. Next, Pachycetidae, Pakistan's whales. And now you're looking at this thing and you're like, where's the whale? You know what I mean? Like this just looks like a guy. Like this looks like a generic mammal. Pachycetus, even older, just as we would expect. Where are they? Indo-Pakistan, just as we would expect. And that's a typo. Not virtually all ocean habitats, 0 ocean habitats. Pachycetids were small, they were tiny. They were 3 to 5 feet long, mostly terrestrial. So they're spending some time in the water, but they're mostly spending time on land. Their limbs are really well adapted to walking, but not running, which is interesting because they've got those big like paws. They've got those big honking webbed paws for walking around and for swimming. Their elbow is mobile, and finally these guys can supinate, but not very much, just a little bit. So they could they could pivot their wrists. They have forward-shaped nostrils or forward-facing nostrils. They have an involucrum, and in fact, their entire inner ear setup is still more similar to cetaceans than it is any other animal that we know of. They have a non-specialized tail and heterodont teeth. Their ankles, just like every subsequent cetacean, they have that artiodactyl double polyastragalus, and they still have that same hoof feature at the tip of each toe. So these guys have little hooves, just like everybody else did. Here's something that's really interesting. For a really long time, we thought that cetaceans descended from a group of animals called mesonychids. Here's the mesonychid that you saw earlier, right? They've got these little hooved digits, and here's Pachycetus over here, who has these little hooved digits and also an astragalus. And the reason I point this out is because until Pachycetus was discovered, these mesonychids were thought to be the last land relative of cetaceans. But when Pachycetus was found with an involucrum that only whales have, with hoofed digits and with an astragalus, whereas Cynonyx over here, a mesonychid, has no involucrum, hoofed digits, and no astragalus, we were like, okay, it's Pachycetus. Like, these guys definitely evolved from, like, if anything, they evolved from this group of animals, from the ones who have the whale ears. But then the question was, okay, but what is Pachycetus? Is Pachycetus a mesonychid? And then we found fossils from this guy here, Diacodexis, who's of course older than Pachycetus is. And Diacodexis has the same type of foot, like the little hooved toes that we see in Pachycetids, and it has an astragalus. It doesn't have that whale inner ear. So what this did for us, if you can just look at those astragali next to one another. It's just dead rare. It allowed us to say that cetaceans are more similar to artiodactyls than they are to mesonychids. And all of these guys are mammals, so they still share a lot, but they form this type of little family tree. There's a cool little part of science, is finding out where we were wrong and changing our minds. Just like all later cetaceans, here's Pachycetus's involucrum. It's this little bulge here that you can see that's marked INV. That's the involucrum, and then of course it's present later in our protocetids and then in modern day whales. Here's another view of the tympanic bulla of Pachycetus versus Adiocetus, one of those neocetes. And Pachycetus had semicircular canals that are, just as we would expect from an evolutionary perspective, more similar to cetaceans than they are to land mammals like galagos, but not identical to them. Right? Like A here is a galago, a little primate. B is Pachycetus. C is a protocetid. And D is a bottlenose dolphin. And change over time, morphologic change over time. And I'll add, this is a cast. Like, this is a CT scan of the inside of a Pachycetus skull that we have, which is really cool. So there's our galagos, there's Myocetus, and then here's our bottlenose dolphins. If we're still doubting Pachycetus having connections to whales because of that inner ear, here is the underwater incus. So Pachycetus is incus over here, one of those inner ear bones, compared to a deer versus basilosaurids, baleen whales, and toothed whales, and it's a dead ringer for the cetacean side of things. It's got this L that's seated on top of a little stoop, whereas in deer, I should say, and other artiodactyls, it's just a regular old L shape. So, Pachycetids are cetaceans. If you'll recall, the primary trait used to define just living cetaceans, the best thing that unites all of them, according to the people who study cetaceans, is this really specialized inner ear, from the involucrum to the semicircular canals, etc. And we can trace the emergence of that modern pattern from terrestrial artiodactyls all the way to aquatic cetaceans. And that's really cool. But what came before that? Rowellids, like Indohyus here, are proposed to be not the ancestor of Pachycetus, but probably similar to what Pachycetus descended from, sort of like in our wolf example. Here's when they lived, so they're not in the right time. Everything else that we've covered checks the boxes for like a classic transitional species. It's morphologically and temporally intermediate. Not Indohyus. Indohyus is too young. These Rowellids are too young. Not by much, as you can see there in the dark purple, but still too young to be like, oh, that's the ancestor. And yet, they're 3 feet long. They're terrestrial. They've got limbs for walking on land. Their elbow is mobile and supination is possible, but limited. They have forward-facing nostrils, and they have an involucrum. They have a non-specialized tail, and they have heterodont teeth. Here's their astragalus compared to Pachycetus, and here is their little toe compared to Myocetus. These guys, at least according to what I was reading by people who study cetaceans, are probably very similar to, if not the direct group, that Pachycetids descend from. Of course, they're not in the right time. They're too young to be the specific group, but I think it's fair to say that Rowellids are probably very similar to what they come from. And these guys are their closest relatives, like in the entire fossil record, is also Pachycetus. Like, it goes both ways. But is there anything else that could specifically tie Indo-Hyas to cetaceans? Because otherwise we could just say, well, what if the involucrum is just found in things other than cetaceans and the rest of the inner ear, which that's a hard sell for me, but more is better. Bones are cool. They're really interesting. This is a cross-section of a human femur. And what you can see is that the middle of the bone is hollow. Really, it's not hollow. It's mostly filled with spongy bone, right, or cancellous bone, whereas the compact bone on the outside is cortical bone. Cortical bone is heavy. Spongy bone is light. This is Indo-Hyas's cortical bone. So this is a cross-section, a CT slice of its femur here in J, and its humerus here in K. So the bone of its thigh and the bone of its upper arm. And you can see that they've got really, really thick cortical bone and not much like spongy bone in the middle. This is cool. And this makes us think that Indo-Hyas probably spent most of its time wading around in the water, because heavier bone lets you sink. So it's probably wading in the water, walking along the bottom of river bodies, probably eating the vegetation down there. And people hear that and they're like, why would we extrapolate that just from the bone? That seems kind of stupid. But the reason is because there's a modern analog for Indo-Hyas. This is a CT scan of the humerus and femur. I think it's the humerus. It might be the tibia included as well here, of a living animal. And that living animal is hippos. These are hippo cross-sections. If you will recall, the closest living relative of modern cetaceans, genetically speaking, is hippos. And the first thing that is clockable as similar to cetaceans in the fossil record is acting like a hippo. That's so cool. That's crazy, especially because Indo-Hyas wasn't discovered until like the 2000s. Just for comparison, here's Indo-Hyas over here. Again, I've got the hippo there for comparison. Below it is Cainotherium, which is a little deer-looking thing, and you can see the cortical bone is a lot less, taking up a lot less space. And the reason is because deer spend a lot of time running, and you don't want really heavy bones if you are running from predators. For comparison, here's a sheep over here too. It's got a similar condition. There's Pachycetus right above it with even more cortical bone and very little medullary bone, or very little, cancellous bone. So putting it all together, when we look at cetacean evolution as a whole, bone density seems to shift within fossil specimens to support the shift from terrestrial to aquatic. So we see more cortical bone to significantly less, and then eventually you get these pock marks in the ambulocetids, and onward, because these guys are regulating their buoyancy instead of walking on the bottom or walking on land at all. Likewise, isotopes come to the rescue here, because turns out what you eat and drink reflects in your bones. It records itself in your bones. We can look at the oxygen content, oxygen 18, our old friend from last time talking about ice cores. We can look at the percentage of oxygen 18 and also of carbon 13 in the bones, in the fossils, the fossil bones themselves, and know whether or not they spent more time in salt water or more time in fresh water, or for that matter, time in water at all. And what's really cool is that in this figure to the left, they have a bunch of living animals to compare it to. So you'll notice that just like we would expect, common dolphins, bottlenose dolphins, striped dolphins, spinner dolphins, pantropical spotted dolphins, lapata dolphins, takushi, Dall's porpoise, killer whales, minke whales, sperm whales, all of these guys track marine. They've got high amounts of oxygen 18. Whereas in our freshwater specimens over here, the only living animals that fall into that group that we sampled here are river dolphins, and all of them fall in the freshwater category. Then we move to our Miocene whales, our neocetes. All of them clock with the marine specimens, as does Indocetus, but Pachycetus, Nalicetus, Ambulocetus, all of these guys are clocking as freshwater, and then Ambulocetus, that one who was bridging the gap between the Pachycetids and the Protocetids, spans the gap, which is cool. That's what you would expect from that's what you would predict from an animal that's going from living in river deltas like a hippo does to being a fully marine animal. And here's another look over here that includes the carbon 13 values. Likewise, brain and body size increase overall in diversity across cetaceans, which is just cool, because cetaceans, as we would expect from evolutionary theory, when a lineage first starts, it takes it a while to diversify. So when we look in the fossil record, if all of these guys lived in the very recent past, we would expect the diversity to be relatively similar as we move through time. But instead, we see some teeny tiny little traces of cetacean looking things, and then over 10 million years, an explosion of them across the world. So I'm going to say that the available evidence that we have suggests cetaceans evolved from terrestrial artiodactyls over the course of about 10 million years. The support for this comes from taxonomy. They nest with the artiodactyls or even-toed ungulates. Ontogeny and anatomy, cetacean development and morphology supports a terrestrial artiodactyl evolutionary history. Paleontology, we see the emergence of the traits that define modern cetaceans in the fossil record over time. In genetics, we can pinpoint specifically the genetic changes for things like loss of hair, loss of limbs, the ability to dive really, really deep, blubber formation, echolocation, things of that nature. We can clock which need to change and how they need to change by comparing modern cetaceans to non-cetacean artiodactyls. I think it's obvious from genetically and from the fossil record. It hasn't been for very long. Like, a lot of this is relatively like past 20 years stuff, but I think that it's pretty clear at this point. I'll say if cetaceans didn't evolve from a terrestrial artiodactyl over 10 million years, it sure looks like they did, right? Like, I sat here and thought, because I was trying to decide whether to include this last slide or not. And I was looking at it and I was like, okay, like let's say this isn't enough. You know what I mean? Like, let's say that this isn't compelling enough. What more could do it? Because to me, every major step is accounted for in the fossil record and in the genetics. So, you know, when you respond to this, I would like to hear from your perspective, if you don't find this compelling, or from anybody out there who maybe doesn't, like, what would count? Because this strikes me as pretty cool and compelling. So if support for cetacean evolution, the arguably the most convoluted, biggest step, I think, for evolutionary theory to overcome, at least in mammals, can be demonstrated, right? From tiny terrestrial artiodactyls to enormous, largest animals that we're even aware of, deep diving, completely different body plan whales of today. Like, what's out of bounds for evolution? Is it other major mammal groups, which we'll talk about next time? Is it birds from non-avian dinosaurs? Is it things like flight? Is it mammals from like reptiles, I'll put in quotes? Maybe tetrapods from lobe-finned fish? Like, maybe losing characteristics is fine. Like, you can get a new body plan if you're taking limbs away, but maybe gaining them is more impossible. Maybe multicellular life from single cellular life. Or maybe it's only humans that are weird. Maybe it's humans from non-human apes that is not possible. But what I would like to do is, over the next 2 lectures and then the remainder of the series for the human stuff, I'd like to go through all of these and try to present a similar case as I've done here for cetaceans. So, to be continued. How do we do on time? Oh, not
Great.
bad.
Yeah, not bad.
Awesome. Sweet.
That was a lot of good information. Thank you.
Did new stuff, cool stuff, maybe? Cetaceans?
A lot of new stuff. About 90% new.
That means I'm doing my job right. Chad is going to be like, I knew all this already. I didn't learn anything new at all. They're going to rain on my parade.
I hope not.
All right. Let me get to our super chats here. Do you want to give any like first thoughts or anything, or do you want to jump right into supers?
Let's go into super chats.
Okay, sweet. Okie dokie. So what we're going to do first is we are going to start with the ones Will pointed out, thankfully, that some people super chatted before the show started. Don't do that anymore, you guys, because if you do that, we'll miss them. Luckily, Will caught it, so I copied and pasted them to the side. That means I cannot highlight them, though, so you're just going to have to take my word for it. The first super chat is from Nethefarius for 50 USD. Nethefarius says, question for Will. If God created a fully grown tree, and I took a core sample from that tree, and I found that it had rings that perfectly matched the surrounding normal trees, would creating the tree with such rings be a deceptive act on God's part?
Okay, this is a really good question, and it's a big super chat, so I want to give you a good answer. So I'm going to try to cover all my bases here. Number one, I want to point out something which I hope is agreed upon by all, but it might not be, which is that deception is not inherently wrong. So when we throw out this word, I think it's important to have an understanding that there is what I would call moral deception versus immoral deception. Um, I I love surprise parties. I've thrown a ton of surprise parties for family and friends. Those involve a lot of deception and even outright lying to pull those off. There's also deception required if you're trying to protect yourself from a situation that's not a good situation. There's a story somewhere online from Ohio State University, which is about me. There was a guy holding students up at gunpoint and stealing their money. We all knew about it, and they said, watch out. And so he got to me one day. And so I had to use deception to get out of that situation. And then I ended up involving the police, which led to his arrest. So I was kind of like hero student of the week. But to answer your question, I do think there would be immoral deception on God's part if he did certain things. I've mentioned this before, and I'll say it again. The most obvious one to me is giving Adam and Eve, if they are real and he created them as adults, giving them fake memories of a childhood and other family members that didn't exist. That to me would be immoral. Your question is a tough one, which is if God created a tree and we cut it open and there were tree rings, would that be immoral deception? And I don't know, possibly. I don't understand trees or tree rings. And so if there's something about tree rings that impact the future growth of the tree, I'm just spitballing here, then it might be required, right? If he makes a tree without any tree rings that would end up just leading that tree to die, then that wouldn't be a good situation. So anyway, I'm kind of torn on that one. And I hope that's a good answer. I'm really trying to think this through and give you what I think is, my best answer. So hopefully that does it. And if not, let me know.
Is it okay, since we saved so much time, is it okay if I ask you a question that's sort of in that same vein?
Oh yeah.
So last time we spoke, when we were talking about Age of the Earth stuff, you mentioned that that you were not of the idea, like that you were of the idea that it would be deceptive to like put fossils in rock that's like that aren't real, like that those animals didn't exist, right?
Mm-hmm. Yes.
So we have fossils that are as old as 550 million years. And it's in rock, right? And we've got those ages mostly from radiometric dating, but I would also say that we have fossils certainly in rocks that we can get really like 10 million plus year old ages just by like deposition rates, like the same rate that Monty Fleming, the same sort of process that he uses to argue that erosion must necessarily like get rid of too much of the North American plate. With controls, we have structures, as I talked about last time, with like the cliffs of Dover and things like that do require a lot of time, and there are fossils in those. So doesn't that put like from your perspective, wouldn't that put a minimum of the Earth of like at least a couple million years?
That's a good question, and I don't know. I wanted to look into limestone, but I didn't have time, and I wanted to see again. I'm going to process everything I'm being taught through what I'm learning, which is kind of a new perspective, but also my old perspective, which I think the big one is a flood, a global flood, Noah's flood. And so I wanted to look through that because Noah's flood can account for the mass deaths, but I don't know if it can account for something that appears to need a lot of time versus a short amount of time. So I just don't know.
Yeah, let's hopefully before our final discussion in like 6 months, you can like circle back, because I'd love to hear your perspective. I had a back and forth. I've had a couple of back and forths with Creationists before on the limestone thing, because it isn't just that it takes a long time, it's that it takes a long time and also like really nice, serene kind of conditions, which in my head I don't associate with catastrophism of a global flood. So I would be really interested in your perspective at the end of this. if you could be like, okay, I think it's got to be at least this old. And even if it's still even if it's still 10,000 years, like, I'd love to hear like, okay, bare minimum, maybe you're not compelled of billions, but maybe you're compelled of like tens of thousands or a couple of million or something like that. I'd be interested.
Okay. For sure.
Okay, I won't do that for everyone. That was particularly good. $20 from Beautiful Universe Curiosity. Again, this is one of the earlier ones. Will, how do I email you? I would like to send you a poem I wrote called Beautiful Universe. The history of life is so much more beautiful than we can comprehend that it gets lost in translation when just learning the basics.
Yes, I'm going to put it into chat right now. Let's make sure there's no typos. That would be bad. Okay.
Somebody else. Yeah, somebody else has looked at it.
It failed to post, probably because it's an email address.
Yes. Let me.
Is there a mod that can fix that, or they could just put it in?
Can you send it to me in the private chat here, and I'll post it really quick.
Okay, thanks.
While you're saying that, we also have from Swimmerfish34 for Canadian dollars. Cheers, Will, from your friendly neighborhood theistic evolutionist and Calvinist with a whale emoji.
That's
all right,
awesome.
Here, let me copy this really quick and putting it into the YouTube chat. Will's email. Enjoy. All right, now I can start putting them on the screen. Great. Holy. Okay. From Studio 102 for $5 Australian. I was so excited to see this live. Was about to start. I did a little dance. Thank you so much, Will and Erica. You guys rock. Thank you very much, Studio. I love a little dance. From No Rum Pundits for $2, cetaceans needed. You think you're real funny with that pun, don't you? I come after you in the night. From Anna the Elephant, 1937 for $5. Is Proboscidean evolution a case study with proximity to cetaceans, or must I wait for it? Yeah, you're going to have to wait for it. We'll cover it at the beginning of next time. We'll do like a couple of quick looks at a couple of other mammal groups, but you'll have to wait. Sorry, Anna the Elephant. I know Anna loves the Proboscideans. From Your Online Moon Fairy for 100 SEK currencies. I feel like that might be, I don't know. Kudos to Will for doing this. Question for Will about science in the Bible. In Job 38, how do you pick science Pleiades? This is from the constellation stuff. Versus poetic imagery like the storehouse of snow.
Yeah, just give me 1 second. I want to look up verse 22, which I'm not familiar with. For those of you who are not understanding where this question came from, I presented Job 38 as evidence of something in the Bible that could not have been known at the time it was written, which is the gravitational attraction of constellations, specifically Pleiades and the Belt of Orion. Yeah, okay, so it's a good question, and I don't know. Just reading this real quick, I don't understand what verse 22 is talking about. And I don't even know if it's poetic imagery either. The reason I bring science into verse 31 is twofold. One is it's recording God as talking. And so I look at God talking in the Bible versus men talking in the Bible differently. Men, for example, can be wrong or be confused or whatever. The second reason is just because of the discovery that was made after the fact. So this is not a situation where someone can go create something after the fact and say, oh, look, we predicted this. Like, there's no doubt in anyone's mind that this has been written for as long as it's been written. And it also happens to match the science. So that's why I thought it was a good argument, and still do, by the way.
I said I was going to look into that one, and like you with the limestone, I did not. I forgot about it.
Yes, well,
not like you with you in the limestone. I did not have time. I forgot, so I still need to do. From Rob Squared 2 for $9.99. Here's a weird thought question for Will. Say someone thought the world was older than what common science says it is. So I don't know, 10 billion years. How would you convince them it's younger using only common science practices?
I don't really fully understand the question. Do you understand what they're getting at? I don't.
I think they're saying, like, from a conventionally scientific perspective, when someone says the Earth is younger than conventional science would say it is, we would say, okay, but, radiometric dating says that it's older, or like the deposition rates implies that it's older, or whatever. But from your perspective, what would you say? Like, I guess they're saying, what would your response be if someone was like, it's actually even older than that? Like, how would you convince them that that perspective isn't sound?
I can't think of why my arguments would be different from the things I mentioned at the beginning, which is the faint young sun paradox, coastal erosion, original biological material, and dinosaur bones. I don't think my answer would be any different.
You gotta let me send you Fuzz's book.
Okay.
From Keegan8633 for $5. Question for Erica. What's your favorite Gorilla song? Great question. I like a lot of them. Honestly, probably Empire Ants. I like Empire Ants a lot. From Jonathan Rodriguez 1253 for $9.99. Bacteria have really diverse metabolisms and have been subject to evolutionary pressures much longer than eukaryotes as a whole other domain of life. We discover novel metabolisms all the time. Yeah, that's really cool. And I believe that. From Patologic for 100 PLN currencies. Remember, class, cetaceans are very slow on land. That is true. They're not very fast runners. From Stephen 251134 for $5 Australian. Great to hear Will has moved his position on age. I think if he continues to look into it, the facts will be persuasive. Keep up the good work. I'm biased, but I do hope that happens, and I'm confident that it will. And
okay.
And I'm gonna pester you in between now. So the next talk, for those of you, it's not up yet because I'm always late on getting it up. 14th of April, and in between now and then, I will pester you with. I'm going to send you Fuzz's book, and I'm going to pester you with faint young sun and stuff, and I'm going to look into this erosion one because I don't know it well enough.
Okay.
We have from Potato Splash 9.99. The only brilliance I care about is what I can pass on to the next generation to build on. What we know about plate tectonics is surely wrong today, but the idea is productive and should be watered. So I guess the idea here is that it's like there's always going to be things that we're going to be wrong about. The goal of science is to be less wrong than we are currently.
Wonder why they say plate tectonics is surely wrong.
I don't know. Maybe it's maybe they mean incomplete. Like maybe we're going to figure out more. Like maybe there are things that can make it slightly slower or slightly faster or change the direction, or you got me. We have from Dopamine for $10. Thank you, Will, for taking this topic seriously and reevaluating your views. I've done it and it's very hard to do. Keep it up. Keep it up, the great work. And Erica, thanks so much for doing the series. My pleasure. I love doing this stuff. From Giant K Pseudo for $10. The willingness to answer honestly, I don't know, is one of the most important steps in skepticism and the search for the truth. I wholeheartedly agree on this, and I think not enough people are comfortable with just saying that. Like Will and I were talking about the some of the medical miracle stuff sort of behind the scenes, and I don't think I think if you presented to me with every medical miracle, I don't think I would be able to give you a great explanation for all of them. I really don't, like a great naturalistic explanation. Saying I don't know is fine. And I said this to Will too, but like that doesn't mean that it's miracles, but it doesn't mean that it doesn't mean that you should immediately whole cloth consider nothing that is unexpected. Sometimes you're going to find new scientific reasons for things too. And by just assuming that it's this without looking into it more in depth, I think we miss out on something there. We have Stephen 251134 for $5 Australian. Could you please do a session where Will provides some of the points of contention ahead of time so Erica can address specific arguments? Mad respect for both. It's tough to find time for these as it is, and they do take up a lot of time. Will and I do talk quite a bit behind the scenes about some of this stuff, and my goal, as I've said a couple of times before, my goal is to, when I repackage these live streams, when I get the time to do so, because I'm just underwater this semester, like the cetaceans, I would like to repackage these videos and have a segment at the beginning where I can kind of address some of Will's contentions, even though some of them are rectified later on in the series. I think that would make it a little bit more comprehensible. But we also do have discussions about this behind the scenes. I hope that's going to be acceptable to you.
I will also add this. I will be sending Erica ahead of time any last issues I have for our last session, so that won't be a surprise to her.
Yeah. Will also offered to do the same for this session, for Age of the Earth stuff. Like, before we had this one, at the tail end of the last one, he was like, I think you said it in an email, but you offered to give me ahead of time the contentions. And I think I said I was fine either way. Like, it doesn't really matter to me. I've heard of these contentions before. Faint Unsung is a little bit, I forget. It's been quite some time since I've looked into it, but erosion and soft tissue, like, I can answer some of these at least, give my best guess on the fly. We have from $10 from the Magic Flutist. Thank you so much for making this happen, and thank you, Will, for putting yourself in the hot seat. As someone who rejected young Earth creationism in my transition to agnosticism, I'm learning a lot. I'm loving that most of you guys are just gassing up the series. From Hoplusek for 129 CZK, I almost said X, CZK dollars, listening to two people of different opinions often feels so pointless, in one ear and out the other. This is different though. Learning happened and I love it. This series, I've said this to Will before too, but like, this series has been awesome for me. I learned a ton of stuff.
Absolutely.
We got Rando from chat for 100 ARS currencies. You think parameciums and humans are related? Is that what college is teaching you? You should get your money back. That's a Kent Hovind quote. But it's harder than you think to make a simplistic cartoon nested hierarchy. From Freak Zero for 199. Sup? Test of super chat. Well, thank you very much. That's very kind of you.
It worked.
It worked. From AI Dames for $5. Erica, thank you for this incredible course that we can enjoy. Will, thank you so much for all you did in the final experiment and for your willingness to learn.
Absolutely.
Again, it's our pleasure. We're having a blast. Well, I am at least. From Aaron Marks 9366 for $10. As a linguist, thank you for including a comparison to phylogeny in language. The spread of languages through human migration has many similarities to biological radiation. Great series, Erica. I wish I could take credit for its use this time, but I just included it because Will found it compelling the first time. It got the point across. And so I was like, okay, how do you feel about the nested hierarchy stuff? That can be a tough concept to grasp sometimes.
Yeah, my big question on my mind is, probably it doesn't have an answer right now, which is just what's, again, hypothetical. What's going to happen if we start sequencing genomes and they don't fit? I don't know. I don't know the answer to that.
I don't either, but I would tell you, and I'll send you a link for this later. We've sequenced way more genomes of, like, complete genomes of animals than you or I am aware of. Like, the last time I checked, I was floored by how many genomes we've sequenced of different species. And also of humans, like of individual humans, to get a better idea of how populations have migrated over time. I'll send you a link to the NCBI browser so you can see, like, okay, which genomes have we completely sequenced? And you have to be kind of careful because, like, for example, just sequencing genomes is really difficult because if you'll recall from like our second lecture, there are a lot of like super repetitive elements. So you might have like ATA a million times. You know, and sequencing that is hard for technology. It's like really, really labor intensive to actually do. So it wasn't actually until 2021 or 2022 that we fully end-to-end every single base pair sequenced the human genome. Like, we sequenced it in 2001, 2002, I think, but we didn't actually fully sequence it until like just now, which is wild. But now it's completely done. Like, we have a complete reference genome, and we have a complete reference genome of quite a few critters now that have some interesting implications. We'll obviously talk a lot more about humans when we get to the human evolution stuff. From Brandy Andy's excellent essays for $5. Fun fact, Will Duffy once reffed a hockey game with the current NHL player Troy Terry of the Anaheim Ducks. Is that true?
So I have no idea how this guy knows this, but that is actually true. Yeah. That's interesting. I'll tell a story. So the first time I reffed Troy Terry, he was a peewee, which means he was 11 or 12 years old. And
Dang.
he was so good. I went up to the coach after the game and I said, "Who is that kid?" And he said, "Oh, that's Troy Terry." And I said, "He's unbelievable." He looked like a mini NHL player at the age of 11 or 12. I went
Wow.
home. I went home and I told my wife, I said, "There's a kid who's going to make it to the NHL for sure." And he did. So he's a great hockey player.
I'll have to ask my husband about that. My husband's like a huge hockey guy. He'll probably know exactly who that. Actually, he's listening, so he'll probably respond in turn here in a minute. That's crazy and funny that they know that.
I have no idea how they know that.
From Isirl 447 I6J for $5. That which is will live. That which is will live. For a second, I thought this is that which is will live. Like Will is live right now. So thank you for clarifying. And thank you for the donation. From Potato Slash for $1.99, making sure Super Chat is working. You guys must have been gagging around about this. I don't know what the deal is about checking on Super Chat, but thank you. We have from Cedarwise for 20 Canadian dollars, Gutsy Gibbon. When some people are shown a contradiction in the Bible, they say it doesn't count if there's any scenario in which both accounts are true, no matter how improbable. Are you doing the same with the faint young son paradox? I hope not. It's been a while since I've looked into the faint young son paradox. I remember when I did, it was back when I was mostly on Reddit, and there was an account, so that would have been like 2019-ish. And I remember when I checked then that there were a number of hypotheses that seemed to address it quite well. So I don't think I'm doing that, but I can't say for sure because I haven't, it's been a minute since I've looked. So I'll look and I will let you know next time because I certainly don't want to be doing that. I don't want to be like giving conventional science a pass for an open problem if it is in fact an open problem that doesn't have a solution. But I'll need to look into that today. I'm just telling you what I remember. And thank you for your donation. From ESRL 447 I6J, again for $5, the only difference between us and everything else is time. Ants think they know. The ants certainly know how to get into my house and clog up my light switch. Also, leafcutter ants have like fungus farms, which is really weird and cool. From Will Gord for $10. Will, if you find yourself convinced that evolution and old Earth are true, how would this affect your perspective of the creation story in the Bible, specifically with regards to Adam and Eve?
So really good question. I think about this a lot, and I just don't know. I just don't know. I know people have dealt with this, and there's books written on it. I just haven't read those books, and so I kind of have to take this one step at a time. And being convinced of evolution and old Earth are going to be the first step before I figure out exactly what to do with the rest.
You mentioned William Lane Craig when you were talking about theistic evolution, which is he's got a very interesting hypothesis for how all of this fits together. I obviously disagree with it, but it's fascinating. We'll definitely touch on it. I think we'll have to. Like it's not one of those things that it's like I don't think there's like it's possible what he points out. Like it doesn't contradict any science that I'm aware of.
Interesting.
But it's not like there's no support for it either, is what I'll say. It's sort of it's in my opinion, it's a bit just so, but we'll get there when we get there.
Yeah, I mainly know him on the theology and philosophy side, so I don't even know what it is. So I'll learn that from you.
He's got okay. I'm gonna give you like a little teaser here. He proposes that Adam and Eve are hominins, but that they're not Homo sapiens. So he's proposing a specific type of hominin. I think the latest has been he proposes Homo heidelbergensis, which like they're a big brained hominin. They use a ton of different tools, et cetera, et cetera. Certainly humanish. Like you wouldn't look at them and be like, that's not a human. Interesting though. Like I find it mostly interesting how he gets around bottlenecks, but again, for later. We have from Darwin, the professional, we have sending encouragement to the best creationist of the year and the modernist ape currently teaching college level evolution class on YouTube. It is our pleasure. I'll speak for both of us. From Spoenge for $5. Thank you both for this. You both agree education is super important. So I'm curious what your, mostly Will's, dang it, opinion is on Kitzmiller versus Dover are.
Yeah. Is this the intelligent design case?
Yes.
I don't know a lot about it. I do think intelligent design should be taught for sure.
I have said this before on various channels, but I do not think it should be. And my main reason is because it lacks a model at present. So I can send this to you later, but you know Michael Behe? Michael Behe had a conversation with Dan over on his channel a couple of years ago. And the channel was mostly on like a state of affairs for like intelligent design per the Discovery Institute, like with where they're at. And Michael Behe in that conversation did like own the fact that at least at present, intelligent design doesn't have like a model. So like it doesn't have a way to test it. It has irreducible complexity, but we have shown in the lab, we'll talk about this with multicellularity later and with the bacterial flagellum in not this next lecture, but the following one, we've shown in the lab like how structures that would meet Behe's definition of irreducibly complex can, and then we've shown that it did evolve, like experimentally speaking. So that makes it hard to say irreducible complexity works as a model boundary for evolution, because if you can show that it can be broken, how do you actually define it? And Behe was like, this is something that we're working on, but it doesn't exist yet. And so what I will say is if intelligent design can generate a model that has predictions that it makes, and can accurately predict things, there should be no problem with teaching it in a science class, but it doesn't have that at present. So that's my issue with it in class.
Do you think the law of biogenesis should be taught in the classroom?
The law of, like life from non-life, because then it's like abiogenesis. We teach it. We teach it historically. So we'll teach like how the law of biogenesis came about. I think that abiogenesis, at least when I was taught it in school, it was taught very candidly in the sense that this is something that secular science has not solved. And so, and of course, I went to a public school, so for high school, so they were very, like you're not teaching religion, but it was very friendly towards the idea. Like it wasn't, my Mrs. Bing wasn't shutting anybody down. So like, I think abiogenesis is one of those things where you can just teach its current limitations as we know it in biology classes or in biochemistry classes. Same with evolution. I honestly, like, people talk about James Tour had an interview with Tucker Carlson recently where he was, James Tour is on my list right now. I'm irritable with him. But he said in this conversation with Tucker Carlson that he thinks evolution should be taught in schools, but that its flaws need to be taught. And I'm all for that if you can point to the flaws that exist. You know what I mean? Like, as far as I know, we teach areas that we're not sure about, right? Like we don't know, for example, the details of exactly how epigenetics works and what epigenetics limitations are. And we teach that limitation. We don't know the exact condition of the last, like in my field, we don't know the condition of the last common ancestor of all African great apes. We have a decent idea. There are some hypotheses, but everybody's arguing. And so when I teach that in class, I'm like, we don't know. This person says this and this person says this. And you can make up your own mind. Same with Naledi burying its dead, but that's another story. So that's my thoughts. We have from Trasvi1 for just $5 Australian, and it's just a donation. Thank you very much. From Where is Waldo, 10 Canadian dollars, just to show some support for this series. Thank you, thank you, thank you. From Ibonham 4388 for $2 Canadian. When will you debate Made by Jim Bob? I was on a stream for 7 hours waiting for Jim Bob to call in and debate me, and he would not do it. But I will debate him anytime. Standing offer to have a conversation about evolution, but he has to agree to talk about the data, right? Like in this show with Will, we can talk about the philosophical stuff, but I've never seen Jim Bob willing to or accurately discuss even natural selection. And I think that's a problem, because if we're going to debate the veracity of evolution, we need to be on the same page about what the mechanisms even are. And I don't think that that's an unfair request. We have from Petite Flower 5259 for $5. The most amazing and compelling part is how multiple independent lines of evidence support and back up the same conclusions. I concur, and that's what I'm trying to show. We'll see if it comes to fruition. From Kevin Moses 4201 for 1,000 Y currencies for Will. Most theistic and atheistic evolutionary scientists can hold their questions about God and agree about evolution being how diversity of life happened. Theism isn't a scientific question.
I'm rereading it 1 second.
Please, by all means.
Yeah. I think what you're saying is true. I will say some theistic evolutionists, I believe, have God intervening at certain points. When you say theism isn't a scientific question, I don't know how to respond to that. As a theist, so it's funny because I get accused of God of the gaps, and I will admit to God of the gaps, but it's even worse. I believe God is the God of everything, not just the gaps. So I don't see how you can have science or philosophy or math or anything else without God. So I don't separate the two.
We have from Mythos Praxis $4.99, just a super sweet donation. Thank you, my friend. We have from Ozzy Damians for $5. The nitty gritty of specific species evolution is my favorite thing to learn. The web of life is so beautiful. Thanks, Will and Erica. Species drive me crazy, but I will talk about them forever. We have Rando from chat for 100 ARS currencies. The moon is getting farther and farther away from Earth. If we go back in time at some point, the moon is basically crashing into Earth, and unless phenomena don't move eternally in the same direction, I would assume is what they're saying. Mm-hmm. This is not true. And the reason is because the rate at which the moon recedes is related to the actual, not just the density of the planet, but the what's the word I'm looking, the positioning of the continents, the tectonic plates themselves. And part of this is because we see this and know this because the moon impacts the oceans to the degree that it causes tidal bulging. It's like a classic. That actually, my favorite figure of tidal bulging comes from Answers in Genesis. I've got a really nice figure for that to explain it. So the moon's recession has not been constant over time, and we know this, right? We can take the current positioning of the continents now, the rate at which the moon is recessing now, the mass of the moon, the mass of the Earth, and we can plug this into models and regress it, knowing how the continents have changed over time, and this is demonstrated. That's my gripe with this. I can send you, Randa from chat, email me and I can send you a paper on this if you'd like. From AmbiZ4233 for $2. This was so cool and so well presented by Erica. I love reading really kind compliments. Thank you. We have from Blackmore 278 for $10. Planetary surface temperature depends on more than the amount of sunlight. Venus is hotter than Mercury due to atmospheric CO2. The sun was larger than it is now as it sheds weight via solar wind. So I think, okay, so this is a faint young sun thing. So what they're saying is the surface of the Earth depends on more than the amount of sunlight. So the surface temperature could theoretically be warmer even if the sun is fainter. Venus is hotter than Mercury due to atmospheric CO2. Okay, I see what's being said here. So basically what they're saying is that the sun could have been fainter, one, because it was larger, and two, because we could have had insulating mechanisms that could have sort of hugged the heat that was present to the surface of the Earth. I think that's what they're saying.
Yeah, I think for this particular thing, again, I don't think there's any solution that everybody's like, yeah, that's a good solution, because otherwise, why wouldn't that be updated online? But I do think uniformitarianism will have to be abandoned for this one.
Quick check. Modern uniformitarianism isn't a thing, right? Like we use actualism. So in this case, people would have to be like, for this instant, you're right. Uniformitarianism would have to be abandoned. But like as a general rule, geologists are okay with being like, obviously volcanoes aren't erupting 24/7. You know what I mean? Like local catastrophe can happen. So, but they would have to invoke a different rate, and there would need to be a mechanism for that. But again, I need to update myself on this. From Mameteor 4003 for $5, highlight of my month. I'm just jealous that Will doesn't have to take exams on this stuff. I was a mean exam writer too. My exams were kind of hard. I believed in my students though, and you know what? They rose to the occasion. One time I wrote an exam that was too difficult, and my direct supervisor, this was when I was teaching the lecture, they're like, you think they're going to be able to handle this? And I was like, I do. They nailed it. They hit the ball out of the park. I mean, the average was like a B-, I think. The next semester, I was told to dumb it down, and so I did, and the grades were worse. So I believe that to some degree, students rise to the occasion. From Mythos Praxis for $9.99, ancient Christians saw nature and scripture as two of the same author. Can evolution be God's divine code for self-sustaining world? Conflict only arising when humans misinterpret the programming or the text?
Yeah, I think human misinterpretation is clearly happening. Just look at all the disagreements. That's one of the things I personally enjoy is more study, more research, trying to figure out what the original authors intended, what God intended. And so there's a lot of possibilities here for sure.
$10 from Danit and the Dinosaur. Danit and the Dinosaur. Got it. Erica, thank you for the series and for all of your videos. Good luck with the PhD. Yeah, thanks. I wish I was able to pursue proper education for paleontology, but you helped so much. Can't wait for Dr. Gutsy Gibbon. The sooner it comes, the better. I had a meeting with my advisor just this afternoon, and I got my fingers crossed for soon. And I'll leave you to determine when soon is. From Joshua Ray Powell for $10, I may have missed something with the Job reference to Orion's belt, but the stars in Orion's belt are not gravitationally bound. They are many light years apart.
Yes. So I'll post the verse in the chat so you can see it. Good question. In Job 38:31, God is quoted as saying, "Can you bind the cluster of the Pleiades or loose the belt of Orion?" Meaning the Pleiades are gravitationally bound and the belt of Orion is not. So you are correct there.
We have $2 from Jazz Jitan. Thank you very much. We have from Reaver 117 for 10 Canadian dollars. I think the most interesting element of cetacean evolution is how they established a niche in a competitive ecosystem using novel attributes evolved on land. For example, efficient access to oxygen. I think the only reason it happened at all is because of when it happened. There were no marine mammals that I'm aware of during the time of the dinosaurs, for example, because there was no space niche-wise. I mean, you think a mammal is going to compete with a mosasaur? You know? So I don't know. I think that timing was key on this. We have from Bill McDonough, 3750 for $5. Erica, Princess Mally raises a good question. Is the skeleton behind you in a timeout? How was it naughty? It is in a timeout. It's in a timeout because it wouldn't behave when I was trying to show the carpals in a Skeptalk call the other day. And also it's in a timeout because I'm mad at it, just in general. We have from Freak Zero for $4.99. If anyone wants to play money on whether Will eventually accept evolution, check it out on Manifold Markets. Friendly gauge of opinion. Will, this one's on you. If you're cool people betting on your perspective change, makes it actually this could be insider trading. Will could be about to make some serious cash on this.
To each their own.
Oh man. Okay. From an honest 8 for $20, could Will explain why the nested hierarchy isn't a sufficient, falsification criterion? We can use it for humans. give a better explanation, not for commonalities, hierarchies, or observations falsifying the macro pattern.
Can you interpret this for me?
Yes, I think what Honest Ape is saying is why the nested hierarchy as presented doesn't work as a falsification criteria, in the sense that we have these genomes, right? Well, we can do this genetically, or we can do this with physical characteristics. Let's do genomes, and let's do primates, for example. Like, primates have all of these specific characteristics that make them unique from all other mammals. So all primates have binocular vision and dexterous grasping hands, and they have nails instead of claws, and big brains, and several characteristics that we could get into the minutia of. Likewise, all the animals that fall into that primate group because they have those characteristics fall into that group genetically. Like, they group there as compared to anywhere else. No strays, they're all in that exact spot. And humans are included in that. And as you move down the list, humans would be included in that too. Down to like the apes, like what I said earlier, orangutans, gorillas, chimps, bonobos. Humans would be thought to be the odd man out, but when you do this genetically, they're not. It's orangutan. So they're asking, wouldn't that be a great falsification criteria, like prior to when we discovered it, is my understanding of this. Because so genetics set up a really awesome test for evolution, because we made all of these predictions about these group relationships before genetics was really invented, before the sequencing technology for DNA, I should say, was invented. And then we were able to sequence it, and it confirmed the overwhelming majority of the placement of these groups. So it was a really good sort of test case. So they're asking why you don't find that compelling, or like what an alternative explanation for that could be, I think.
Okay, I think if it was laid out clearly for me, I think it's a sufficient falsification criterion. I'm a little bit confused on how we can look at genetics and determine which one is younger versus older. So I haven't heard that yet. And I also want to know, again, no one should fear this. Creationists shouldn't fear this, and evolutionists shouldn't fear this. What are we expecting to see going forward, and what would upset the apple cart?
Yeah, I think that's fair. And and we're going to be sort of touching, I'm going to re-highlight this same thing a lot as to like, okay, what did we expect before we tested the genetics, and then what hashed out? What were these relationships on physical characteristics, and then where did that land us? Because I agree, I think it's a great falsification criterion.
For sure.
As for, you said you were confused on something. You were a bit confused on
age.
Age. Molecular clock. I will intentionally, because I could do this on the fly, but I think it's better with diagrams. So, I'm like a visual person. I'll include an explanation of the molecular clock in the next lecture. I'm going to write that down so I remember. In the meantime, because it's important, and I should have included it here. I thought it was going to take longer than it did, and then we actually just blew right through it, which I was glad to see. From Queenie Rex for $10, I just want to say you guys are awesome. I'm loving the series. Cetaceans are my favorite mammals. This one was super fun. They are some of my favorite mammals now. They are way cooler than I thought they were. Molecular clock. Okay, now I won't forget. It's on the list. Next, from Omega Croc 2928 for $9.99, modern cladistics and phylogeny is shaped by genetics in many cases. Several historic taxonomic groups have been rendered invalid in part or even largely due to genealogy. This is true. So for instance, like we didn't know where to place some of these disparate groups that don't have that many physical characteristics to assess. Like aardvarks are really, really different from all other Afrotherians. And so we didn't really know where to put them. Like there's just nothing close to them to compare them to. Like it's easy to say like wolves and dogs are related, and like coyotes are probably similar. And if you find a fossil dog, it's going to fall pretty closely within that group. But in the case of aardvarks or like pangolins, for example, they're like these really weird oddballs that seem to have been kind of on their own for quite some time. And it's true that genetics cleared a lot of that up as far as like who pangolins are most closely related to, who aardvarks are most closely related to, with no close living similar groups. Concede that for sure. We have from Joshua Ryan Powell for $5, also way to go. That was a tour de force. You clearly put a ton of work into that. Very impressive. This presentation is the one I've probably put the most hours into preparing. I will say that. Just because cetaceans aren't the area that I formally study, and I wanted to do it justice, so I had to read a lot about it. I loved it, but hopefully I'm gonna be on easy. The second we get to primates, I'm gonna be on easy street. Will, I'm gonna not have to do any prep for this at all. From Rando from chat for 100 $. Question for Erica: if evolution is so cute, so cool, why is it the worst Dragon Ball movie ever? Checkmate atheist. I have no counter for this because you checkmated me on this. Evolution destroyed. Dragon Ball Evolution sucks. It's so bad. From Baka for $5. Thank you both again for continuing the series. You've been gibbon, pun, hilarious. You're going to get banned for that. A lot of new knowledge, and I will definitely watch the next stream. Luke, if you're listening, ban this user. I don't want to see any of that anymore. From Earthly Skeptic for $5, great transitional fossils lesson. Will evolution supported by fossils and observation that blowhole in dolphin embryos migrates from the nose to the head. I saw this and I forgot to put this in. This is also true. When they're developing in utero, just like their little hind limb buds reabsorb, initially the development of the nasal opening, the nasal aperture, starts at the tip, and then it sort of shifts as the rest of the cranium develops. It's cool. There's some cool, what's the word I'm looking for? In progress stills, plates maybe? There's some time lapses or something out there about it. $9.99 from Plot Twist Dad. Speaking of the Dover trial, quick shout out to Stated Clearly, YouTube channel with John Perry that just started an animated series of videos on the evolution of the bacterial flagellum. Okay, I was going to bring this up to you actually, Will. So you've probably seen John Perry's videos before. He does like these animated sort of educational videos.
Yes,
and recently he's been doing this video on everything we know about the evolution of the bacterial flagellum with Martyr Every Day, I think, with a YouTuber who I believe had expressed, yeah, that's the one. Yeah, have you been watching it?
No, it's in my to watch list.
Okay, sweet. I've only seen the first episode, but I know the guy that he's done it with had posted a video previously where he had sort of expressed, I don't know that he expressed doubt of the evolution of the bacterial flagellum, but it was sort of like a skepticism for its naturalistic origin. So I saw it and I was like, this is just like what me and Will are doing. It's like two people who are like having a conversation about evolution. So I don't know. I'm excited to watch the rest of it. It looks cool. We have from the weirdo CKA Evan for $5. One of my favorite compliments was someone telling me, we're Jews, we're supposed to wrestle with God. Hope you can embrace Christianity's Jewish roots, Will. I think he's already engaged in that now, honestly.
Oh yeah, for sure.
Awesome. From Curbstains for $5, props to Will for doing this from a former YEC. You are just flush with these particular types of compliments. From Design Song 1 for $5, food for thought. If evolution is true, the Flintstones are more likely than Adam and Eve's story. I don't get this. Food for thought. If evolution is true, the Flintstones is more likely than the Design Song. You're going to have to clarify that. I don't know what you're saying here. We have from Mythos Praxis for $4.99. I'm a biology undergrad. Even in high school, you never touched, I guess, biogenesis, but we are taught hot primordial soup, that life cannot come from non-life. This is very difficult for me to discern.
I tried to as well. I couldn't.
I am going to go with that you were taught good stuff. Let's go with that. I hope your biology teacher was good. From Misty 3252 for $10. Erica, great job as usual. Will, I appreciate your curiosity and intelligence. Gibbons are the best apes. That is so true. Gibbons are the best apes. They're the cutest. From Hang Fashy 6024 for $5. Guts a gibbon. If someone was building a collection of skulls that showed human evolution, which ones would you definitely have 3D printed to show students? That's exactly what this progression is meant to do in the fewest amount of steps possible. So you're going to want to start with something chimp-esque. I'd probably go with Paralopithecus catalonicus, then do Sahelanthropus chidensis. That's easily printable. You need Ardipithecus ramidus to show the reduction of the canine teeth. You need Australopithecus anamensis if you can get a hold of it. The MRD cranium is tough. It's not necessary if you can't, but it's a good one to have. You need a composite Australopithecus afarensis, probably Mrs. Ples, your STS5 for Australopithecus africanus. Homo habilis, that's KNMER 1813. Then you want KNMER 1470, which is the Rudolfensis cranium. Then I would go with one of the Dmanisi crania, probably skull 3. Then I would do Turconaboy, because Turconaboy is awesome. That's WT, or not WT 15,000. That's no, it might be WT 15,000. I think it might be. Don't quote me on that though. Then do Peking man, Java man. Then you need a Neanderthal. I have La Ferraci behind me, and then modern Homo sapiens. And you might do like a primitive Homo sapiens too, like an early Homo sapiens. If you could get your hands on Jebel Irhoud, that'd be great. But those are a tough sell. We've got Hawks X for $10 Great stuff as usual. So Erica, how can you explain how, when, and why did we become apes without fur? I'm assuming you just mean like, why do we think hair loss happened? In humans, it looks like it corresponds pretty tightly with running, and running is really, really hard to do without overheating. See the cheetah, for example, they overheat really easily because it's just rapid bursts of energy. And overheating is the biggest threat for predator and prey animals when they're exhausting a lot of energy. Humans, weirdly, are like really, really good at thermoregulation because we sweat. And sweating is really hard to do if you're covered in hair, which is why most hair-covered animals sweat primarily around their pads and why they pant. So in all likelihood, the fur loss came about the same time as our really long limbs. So probably early Homo erectus is the best guess. We have Carson Reed Davis for $10. Will, I'm a fan of saying I don't know, but I'm worried that if the smallest exception always precludes understandings, then mountains of evidence become meaningless. Weight of evidence matters.
Yeah, I agree. I tried to show you that I'm being honest and objective here by saying that the scale seems to tip toward old Earth. Again, we're going to talk about this later. I don't know when, but the dinosaur soft tissue is big for me.
I have, and I want to pick your brain on this as well. I want to know what you think about the experiments that show how long it can last, because I know like, we didn't get a chance to touch on that, but I find those very compelling. And for me to find those less compelling, I'll just tell you what I need to find soft tissue convincing as support for young Earth would be, it would need to be a specific type of soft tissue that these mechanisms wouldn't work for. So when we talk about that, that's how you're going to pull me over.
Okay.
From Kevin Moses 4201 for 1,000 Japanese yen. Sorry, thank you. Will, I just meant to say science is limited to understanding the natural world, and we have no way to investigate the supernatural, so the existence of God is outside of the scope.
Okay, so here's the problem. We can't choose one thing, one field, if you will, and say that that's the only field that we can utilize to find truth. So there's a common theme I'm getting when I read comments and super chats like this, which is that science is either the only way to find truth or the best way to find truth. Both of those beliefs are philosophical, so you can't do science without philosophy. And I think we should just be trying to figure out truth in general. And we use different methods for doing that. In terms of the supernatural, we're looking for something that cannot be explained through natural means. And there's plenty of examples of that. So just to put a period on this here, I'm not trying to go through life only using science. That seems silly to me.
This is similar to how I teach it. I will say that, Kevin Moses, like, I think I'm on the same page with you, but that's sort of neither here nor there for this particular conversation. From Aussie Damians for $2, have you seen video game Evo Sims, Will?
I think the last video game I played was Duck Hunt.
No.
I don't do video games. And I cheated. I put the gun right up to the glass on the TV. That's how old I am. The TV was made out of glass.
I'm changing the subject before I get too irritated. From Mythos Praxis, sorry, my autocorrect is bad. I'm a bio undergrad, and even in high school now, we're taught that we don't 100%, we don't know 100% about abiogenesis, but primordial soup is taught biomolecular stuff. I think that's fair. I think as long as you're like, this is an open, I think it should be an open question because it's an open area of research. I happen to think there are a bunch of interesting and compelling models, but I don't know enough about biochemistry to say which one I think is like the best one. From Nathan Runyon, 8591 for $10. Will, I read this one. I disagree with you here, Nathan. I don't think that that's what Will was saying, and I think he was clear about that at the end, but I'll let him answer this. Will, based on your opening remarks, it seems like you're still on the fence about an old Earth. Have you changed your mind, however, specifically on the validity of radiometric dating?
Okay, so on radiometric dating, I met with John, who you guys might recall has a YouTube channel called Age of Rocks. I met with him multiple times, and he is an absolute wealth of information.
He's a beast.
He's amazing. And he taught me things that were mind-blowing. I now barely understand radiometric dating.
So I feel about it too.
And I feel like I've learned so much. And it's all amazing. When he described for me how this works, all I could keep thinking is people are amazing. People are amazing. Like the fact that we even have machines that can do this is just mind-blowing to me. Have I changed my mind on the validity of radiometric dating? Here's the problem. I was, and John was frustrated with me, probably. He didn't show it, but I was obsessed with carbon-14 dating. That's all I wanted to talk about. And so he and I would talk for like an hour and a half, 2 hours, and then I would be fried just on carbon-14. Then we met again, and I'm like, all right, I got some more questions about carbon-14, and then here we are, hour and a half, 2 hours in, I'm fried. So we haven't moved past it. What he did teach me is that carbon-14 dating is probably the least accurate of all of them. And there's issues, right? A paper came up in my Google News feed in the last 2 weeks where they actually now are saying that tree rings are wrong based on carbon dating because the carbon dating is longer than the tree rings. And so I mentioned that to John. I haven't read it. He said he saw it, but hasn't read it either. So I don't know what it all means, but that's something brand new that just happened in the last couple of weeks. I could hear it.
Yeah, please. I would love to look at that. And I would find that shocking because that would mean like 3 independent things had to be wrong. The carbon dating would have to be wrong. The tree rings would have to be wrong. And the varves would have to be wrong. And the ice cores would have to be wrong. If I had to guess, knowing nothing about this, I would bet it's talking about a specific instance where one or the other with either tree rings or carbon dating without a third or fourth method to cross check it was wrong. But I want to read that because that sounds interesting.
Okay.
I will say too, one thing that I'm really interested to hear your thoughts on after you've had a chance to digest it is like Mount Vesuvius and Pompeii, like these other methods that we have of radiometric dating that like match the historical records that we have from human history. I think that's like one of the most compelling cases for radiometric dating.
Yeah. So what I was going to say is that I'm not as excited about carbon-14 dating anymore just because I realize the limitations. I now understand, why am I forgetting the term? Contamination way better. That has been presented to me. I'm not going to point fingers. That's been presented to me as like poor, sloppy science work. And where it's like, oh, they didn't wash their hands or they didn't, keep the specimen clean. And it's like, no, that's not it. I now understand contamination better in the world of carbon-14 dating. I can't explain this, but the way they're able to do the dating of carbon-14 has to do with electricity. And so just the fact that the machine has electricity running through it creates quote unquote contamination because it's reading something that's not actually there.
It's like
So I thought
picking up on background. I see.
yeah, I thought it was shoddy science, and that's not it at all. So
Mm.
anyway, I'm gonna continue to look into this, and I'm gonna meet with John again if he's willing. By the way, I just shared my screen if you want to show that. That's the paper that I have not read.
Radio.
That people can look into. But I am excited to get into more radiometric dating stuff because I want to learn about the other ones as well. And again, I'm very tempted. At this point, I probably won't do it, but I am very tempted to do some sort of a final experiment for Age of the Earth. So that's where I'm at on radiometric dating.
Sick. Awesome. I'm very glad to hear that. And I'm glad, I mean, I'm not going to put words in your mouth. My impression is that you find it more compelling than you did previously as
For
a
sure.
method for dating.
Yeah, for sure.
Okay, we have from Freako 499. We have to clarify my previous super chat. You don't have to bet real money on Beautiful Market. It's a play. It's play money, but you can bet with real money. Gotcha. Okay, from Steve Van Winkle for $10. If Erica was my science teacher in high school, her joy for science would have had me stay in the science instead of playing World of Warcraft for 20 years. My goal is that's why I'm on the internet, because I want everybody to find it as interesting as I do, because I think it rocks, and I want as many people to have access to science as possible. I hate that bad science teachers sometimes, or ivory tower science sometimes, keeps people from getting excited about this stuff, and I think it rocks. Okay, from Virgil Uncle for $4.99. Absolutely love this series. Thank you both for all the work you put in. Silly question for Erica. What Miocene ape would make the best vamp? Dendropithecus, for sure. Or Wrangler. Wranglopithecus also has these big, buccolingually compressed canines. Freaky. From MMedio 4003 for $5. Another question. Any new thoughts on what a final experiment for evolution would look like? Since Will mentioned it, I'm curious where his mind went. I'm going to let you read this, and I'm going to look at this paper really quick.
Okay, yeah, I haven't spent time on this because I know if I come up with a really good one, I'll do it. So it's a little bit makes me nervous, but I'll tell you two things that came to mind. Young Earth creationists make a big deal about carbon-14 being found in diamonds. And there's a misunderstanding on the Young Earth side, and there's a misunderstanding of people who are not Young Earth. The misunderstanding on the Young Earth side is that this carbon-14 is, shows that the diamonds are really, really young. From what I learned from John, the age that they're getting is so small, it's practically irrelevant. The misunderstanding from the anti-Young Earth side is that Young Earth creationists think diamonds are from organic material, and that's not true either. One idea I had was to get a diamond, which we find, in situ, if that's the right term to use for a diamond, and we cut it up into like 5 pieces, and we send those to 5 different labs, and we live stream the results of carbon dating it. If the ages are all different, which is what should happen, then that's background noise, aka contamination from their particular machines. If it's all the same, that would mean that the carbon-14 is from the diamond. One idea I had. Second idea I had, because of my time in the Hell Creek Formation looking for dinosaur bones in Montana, is there's a lot of coal inside this dinosaur layer. And so one idea I had was to take a core of dirt and sediment and that has coal in it and protect it from the elements, and carbon-14 date the coal, because that is organic material. And I believe the mainstream thought is that coal is many millions of years old. So those are the only two things that came to my mind, but I haven't spent a lot of time thinking about it.
We have from this is goofy. We have from Drumming Otaku for $5. Whose young son fainted? Should we be worried? I'm implementing a universal pun ban if you guys aren't careful. For Potato Splash for $1.99, I've got money to donate towards an old Earth final experiment. I'm assuming that's old Earth.
Email me. We
have from the weirdo KCA Evan for $2. I'm cheering Will's will to wrestle with belief. We have from Cedar Wise for $5 Canadian. Will, I really appreciate how you approach challenging questions in good faith, not dismissing, but working towards understanding and engaging. Rare and cool. Rare and cool is right. We have from Omega Croc 2928 for $4.99. Avian respiratory system on top should be called dinosaurian respiratory system. We will be talking about that next time on dinosaurs. From Ian McHenry, okay, 62.88 for $5. This has to be the peak of online discourse. Props to Erica for incredible presentations and to Will for his openness and insatiable curiosity. Yes, concur and thank you, and hopefully that will continue to be the case. From Majari Teldani for $10. Will, what would you require to move from I don't know? Do you need every potential outlier like red blood cells explained? When does the weight of the evidence tilt to where outliers don't block you?
This is a fair question. I experienced this with Flat Earth, to where they would just hang on to an outlier and say, I'm not going to accept the weight of the evidence. There's two issues here, which I have to separate in my mind, even though they're a little bit related, which is age of the Earth and evolution, right? On the age of the Earth, again, we're going to talk about this more, and I'm going to do my homework. The dinosaur soft tissue one has always fascinated me. And if you recall, I was involved at not the very beginning, but close to the beginning in raising money to give to Jack Horner and Mary Schweitzer to do a carbon-14 test on the soft tissue that they found. And that was denied because they didn't want creationists to have that data. So that's really affected me emotionally. And again, I mentioned this before. I'll get this paper to everybody, but a friend of mine who is a young earth creationist just had a paper published in a secular journal. They gave him the cover, which is pretty cool. I know that is a big deal on dinosaur soft tissue. And he's claiming it's a pretty significant deal that what they discovered and found. So I got to get my ducks in a row, but that's the big one for me. I think if that one went away, I would be okay rejecting young earth.
Yeah. Then I got my work cut out for me. And let's organize behind the scenes and maybe we can exchange some papers. And at some point as we move in these, we can have like a little exchange or something. I don't know. We can work something out, or we can just keep going behind the scenes. I don't want to, you're already committing a lot of time to this, so I don't want to suck too much, but I do think that this is worthy. If that's a big linchpin, I think that that's worth investigating. And I'll add, just as an aside, I can appreciate here, especially hearing you, because you've said you told the story last time too, and I heard you talk about it with PhD Tony as well, and I can appreciate that it sticks in your mind because it feels weird that you would offer to radiocarbon date something like this and be rejected because of how creationists could spin it. And it bugs me a little bit that Horner didn't just explain, kind of like how John did, like how radiocarbon dating actually works, and like why, instead of saying this is we could do this, but the results won't mean very much, he said we're not going to do it because of how the creationists will spin it. And I can understand maybe he gets a lot of people, maybe him and Mary Schweitzer get a lot of people on their case about this. I know Mary's talked about she gets a lot of people reaching out to her on this topic in particular, so maybe that's part of it. But I can understand from your perspective why that would feel suspicious and weird, instead of just explaining the situation, just saying no, we don't want, because the information might be misused. And from your perspective, the way that that's being read is we might get results that we don't like. And so I get your reticence there, and I think that's why it's worth digging into this a little bit deeper. So hopefully we can find some time to do that. Okay, we have from Blackmore 278, doing my work for me for $10. The UCSC genome browser currently lists 50,037 genome assemblies. The multi-track species view of genes on UCSC really drives home that all organisms are related, and it's a great resource. I'm going to send this to Will because the genome browser is a really cool tool. I will say, I usually, when I'm doing this, I like to go to the papers and make sure that when we're talking complete genome, we're talking complete genome, because the completeness of the genome in coding regions is a different thing than the completeness of the genome overall. Like we've had the complete coding region for the human genome since 2002, but that makes up like 2% of the genome. Like there's 98%, and that stuff, I would argue that most of that isn't doing important stuff, but some of it absolutely is. And so we have to have the whole thing sequenced if we really want to account for, if we want to say the full thing is done. So I would say agreed, probably some of those are fully complete since we've had the human one for a while, but be careful when you're looking at it. Okay. From Seawise for $5 Canadian, Cutsy Gibbon, could what happened with pachyderms be an answer to Will's question about what would happen if future gene sequencing doesn't match expectations? Potentially, and we're going to talk about that next time when we talk about whale evolution, because that's an interesting case study. From Moth and Man 6313 for $4.99, Will. Okay, I'm going to do my best here. Safaria has a translation of the Hebrew Torah. In it, Genesis 1 and 2 are contradictory stories of creation. Thoughts on this? Thank you both.
I have never heard of Safaria, and if it's a translation of the Hebrew, I'm not sure why it's being focused on rather than just focusing on the Hebrew. So I'm not aware of contradictory stories in Genesis 1 and 2. So don't really have any thoughts past that.
We have from Anony a bunch of numbers for $10 Australian. Daughter and I have been having fun playing Metazoa and Metaflora today. Perhaps Will might benefit, at least have some fun by wasting some time deducing species from hierarchical relationships. That's a fun thing to do. That's a fun thing to do. I don't know. Maybe I'll include some of that like built into the next presentation because it is fun. From Baka Baka for $2. Apologies on the bad puns. You're not sorry. I know you're not. Thank you both. Okay. From Bill McDonough, $3.950 for $5. This one must have just come in. Will, don't let Jack Horner being rude sour you on stuff. Horner's done great work, but he's kind of a jerk. That's part of it too.
Yeah.
We have from greater than 5054 for $5. You should focus on zircons for your final experiment. Zircons can't be contaminated by lead. That sounds cool to me, but I don't know enough about zircons to say.
Yeah, I don't either.
What I will say is that in the background, I pulled up this paper, and this paper is not saying that our radiometric dates are like BS. This paper is called the title kind of says it all. Radiocarbon dating opens up new frontiers in the study of tree longevity, insights from angiosperm trees. And from the abstract here, and I'll send you I have the PDF, so if you want to see it, you can. The explainer actually does a pretty good job. But basically what they're saying here is that angiosperm trees actually live quite a bit longer than we've previously given them credit for, which allows them to be used in correspondence with other methods in more scenarios than we'd previously appreciated, is my current understanding of this. Hold on just a sec. There's a line here that might help. Hold on. Where is it? Yeah, we present the most up-to-date estimates of maximum longevity for 42 arboreal angiosperm trees. Is that all of them? Okay, based on literature review, let's see. Radiocarbon results indicate ages of 400 to 500 are common features of many broadleaf species, with exceptional old ages being restricted to a minority of individual trees. Our results underscore the need for an increased effort to locate old and ancient trees and investigate their role in ecosystem ecology and ensure their protection. So this is not a radiocarbon is BS. We could probably use radiocarbon further back than we thought for some of these trees, I think. Okay, but I'll look into it more and we can rejoin on this. And we'll get John's perspective because John will actually know what they're doing. John will be like, oh, and then they did this and it has to do with this calibration curve, and then we'll know what's happening. Folks, thank you so much for being here tonight. The next of these shows is going to be this same time, 6 p.m. CST on the 14th of April. That's what I have on my calendar right now. If we change it, we'll let you know. It's going to be similar to this one in that, but instead of sort of that intro, we're going to have several lineages that we're going to investigate in the long term. Will, do you have any closing words before we get on out of here?
Yeah, I think that was the wrong date.
Is it the wrong date? I thought it was the 14th. That's what I have on my calendar. What is it?
I have the 15th.
Okay, I must have written it down wrong.
Okay,
It must be on me. Okay, it's the 15th, not the 14th. It's the 15th. I'll change that on my calendar. and I'll get that up as soon as possible. Okay, my goober mode out of the way. Anything else that you want to say?
Yeah, I'll just tell the quick story of what I found in the Hell Creek
Oh,
Formation.
yes. Yes, I wanted to hear that. Yeah, what did you find?
All right. So as I mentioned, when you find even the smallest part of a bone, you just get excited because you don't know what it could be nothing, or it could be the next Sue, a complete T-Rex.
Yeah.
It turned out to be nothing, but there was something behind it. So it's painstaking to do this work. You have paintbrushes, you have toothbrushes, and you have to be so careful, or otherwise it can just disintegrate. Behind it was a T-Rex tooth.
Oh, that's wild.
And they didn't want me to get it out because it's incredibly rare.
And it's probably fragile, maybe.
I said, just trust me, I'm going to take my time. And I spent hours on this tooth, hours, to make sure I didn't do any damage to it. I got the T-Rex tooth out and they immediately confiscated it.
No.
And I'm sitting there like, what's going on? And they said, sorry, Will, this is going in a museum. And I said, why? And they said, they're very valuable. And I said, well, how much are they? And they said, they're worth $2,000 an inch. And this is
an inch.
Yes.
Wow.
And my tooth was almost 2 inches long.
Wow.
So I went to them and said, okay, I got my checkbook out and I said, I'm your buyer, name your price. And so I purchased the T-Rex tooth.
Do you have it still?
So I have a T-Rex tooth from the Hell Creek Formation that I found, which means my eyes were the first eyes to ever look at that tooth.
Do you still have it?
I do.
Can I see it?
Yeah, I don't have it with me right now, but I'll bring it next time.
Yeah. Bring it next time. I really, really want to see it. I really want to see it. That's incredible. Okay, get ready, because I'm going to say anybody who's doing T-Rex work, email Will if you want his permission to get his measurements on that thing. I know Will is an open science guy because of Final Experiment. So with that incredible climax out of the way, thank you so much for being here, everybody. This, as always, is a blast. I always get so nervous leading up to these because I'm always like, okay, well, then what if I say 1,000 things that are accidentally wrong? And then I always have an absolute ball. Thank you for being here. We'll see you next time on the 15th. That's it. Bye-bye. Ending the stream.